I'm going to introduce our first speaker, Jonathan. Jonathan is the UCD Brendan McGonigal Professor of Pediatric Molecular Hematology in UCD. Jonathan Owen, he's also honorary consultant pediatric hematologist at the Cromlins Hospital. Jonathan was trained as a hematologist here in Ireland, but afterwards he decided to start a research career, moved to France and then to the UK to UCL. And I think he had a great career there. Nonetheless, he decided to return back to Ireland in 2018 and established his own research group. And Jonathan, you're very welcome for your talk here.
That's great. Thank you very much, Ava. I'm just going to share my screen. There you go. So hopefully you can see that and hear me. So thanks again for the invitation. My only disappointment is that once again I've been deprived of a trip to Galway, but I'm presenting here from a rather dreary Dublin, looking out the window there. But it is what it is. So I'll just change my view because I can't. Yeah, that's better. So my talk today is basically a summary of a review we wrote very recently for the FEBS journal that has just been accepted, and it's up on the website, although it's still in PDF format. It will be in more glossy form in a couple of weeks' time. So it's really all about how we can use the roadmap of normal blood cell development for thinking about leukemia biology. Now, this is not a new concept, but we think it's timely to revisit this because there's a lot of new computational tools that have been developed in parallel with high throughput genomic sequencing approaches, for example. So that's really what it's all about. And this is the graphical abstract for the review, which was designed by the first author, Thomas Lefebvre. And I think he did a very nice job of translating my kind of vague directions of what to represent in this picture, and has made a very nice image showing the kind of way cells navigate through hematopoiesis. Also the fact that things can go wrong occasionally, but now that we have a toolbox to maybe deal with these things or think about things a little differently. And I was also quite happy, he's quite young, so I was worried he wouldn't know what a roadmap is, but thankfully that's still part of the collective knowledge. So, I don't seem to be moving. Are my slides moving for?
No, stuck on the road.
That's it. To call the AA hand. I'll just share again, see what happens. Okay, it's moving now, yeah. Is that moving for yourselves?
Yep.
Okay, sorry about that. Okay, so just to bring this back to normal blood cell development. So most of you will know that normal blood cell development takes place in the bone marrow in humans in postnatal life. T cell development takes place in the thymus. And very briefly, what happens is you start with these multipotent hematopoietic stem cells, and then there's a chain of differentiation whereby cells become less pluripotent and more committed to a particular lineage until you end up with these mature cells, so mature red cells, white cells, platelets, etc. And leukemia is really a disorder of normal hematopoiesis. So something goes wrong along the way. And really the central problem is this block in differentiation. So we do have, of course, these extra oncogenic mutations that arise in more classical cancer sense, but the underlying problem is that there's an arrest of differentiation. And we know that if we can overcome that arrest in differentiation, then we can do something about the leukemia. So the important thing to retain from this schema of hematopoiesis, so this has changed a lot over the past 20 years, and we know there's quite an old idea now that you have this lymphoid-myeloid dichotomy. So early on you get the separation into lymphoid and myeloid lineages, And we know this is not the case anymore. So we know this from studies in vitro, taking various hematopoietic precursors and culturing them in different conditions, or in transplantation experiments in mice. We know that a lot of progenitors retain multipotency even at a relatively late stage of development. So you can see some of these potentials here, for example, in the multi-lymphoid progenitor, which still retains quite a bit of myeloid capacity. And this is kept even quite late. So these cells, for example, which we'll mention a bit, early thymic precursors. So even when the progenitors arrive in the thymus, they still retain myeloid potential. So you can imagine that a differentiation block for some of these pluripotent stages might have characteristics of lymphoid cells or myeloid cells, or both, or even neither sometimes. So this is the outline of my talk. I'm going to speak fairly briefly about the practical challenges we have in leukemia diagnosis at the moment, with specific reference to this T myeloid space, the immature T myeloid space. Very brief discussion of the molecular landscape of these leukemias, and then I'll be spending most of the time talking about how we might use new technologies to interrogate some of the biological processes a bit differently. Okay, so current practical challenges. So of course, most of you will know, but just for the non-clinicians, acute leukemia classification is still effectively based on phenotypic resemblance to a normal blood cell precursor. So there is increasing incorporation of some of the genetic alterations that are present in these leukemia cells, but it's really pretty much all down to immunophenotype when you class a leukemia. And it still relies for practical purposes on this idea of the myeloid lymphoid dichotomy, which no longer pertains. And this is for practical reasons, of course. So treatments for AML and ALL have developed in different ways, and the decision, obviously a practical decision, has to be made how to treat the patient. So based on the immunophenotype that is obtained at diagnosis, leukemias are classed as acute myeloid leukemia, AML, acute lymphoblastic leukemia, B or T, and then you have some that don't fit these categories. So under the WHO classification, these are termed acute leukemias of ambiguous lineage, and can be either acute undifferentiated leukemias, where they don't express lineage defining antigens, or mixed phenotype acute leukemias, which may or may not be associated with other genetic alterations. So these are the broad categories. And then you also have a kind of wide gray area with so-called aberrant antigen expression. So there's kind of a range of differences you can get here, but just to particularly mention the early thymic precursor subset of ALL, ETP, which is a very distinct immunophenotype, which I'll show you in a slide or two. So you can then superimpose that kind of idea on normal blood cell development and think, well, ALL, this relates to kind of this portion of hematopoiesis. Something goes wrong here, you get an ALL. AML relates to kind of this portion of hematopoiesis. Something goes wrong, you get an AML. But again, these progenitors have oligo or multipotent capacity even at a late stage. And just to stress that, I'm maybe laboring the point, but even, for example, the ETPs and obviously all these cells in here have lots of different genetic programs activated that relate to differentiation. So in terms of classifying, I have a couple of slides on the main classification systems for illustration, really. So you know that in the WHO classification, the lineage assignment is based on expression of a very limited number of antigens, really, especially MPO for the myeloid lineage, CD3 for the T cell lineage, and CD19 for the B lineage. No specific threshold of positivity, so there's recommendations that staining, for example, of CD19 or CD3 should be at least as intense as the normal counterpart, but this does mean there is a range of expression and maybe an element of subjectivity into how these lineages are assigned. The other main classification system is the EGIL, so the European Group for Immunological Classification of Leukemias. And this is a very useful classification system in general because it really refers to the normal pattern of antigen expression in different blood cell subtypes. And the way their lineage assignment works is that there's different levels of positivity or scoring applied to different markers depending on how lineage restricted they are, let's say, or how lineage promiscuous they are. So you get many more points for having CD3 to be a T cell lineage than, for example, CD7, which is very commonly expressed in AML as well, as you know. The EGIL do define a specific level of positivity, so it's compared to an isotype control, at least 20% positivity for surface markers, at least 10% for cytoplasmic markers, and they define biphenotypic or bilineal acute leukemia. So this is a distinction that doesn't happen in the WHO classification, as having at least a score of 2 in 2 lineage columns. So basically, to be defined as any of these lineages, you need a score of at least 2 through either having 2 various combinations of markers. And just to briefly mention, this is the last immunophenotyping slide, but within these criteria, the ETP subgroup, just to highlight this immunophenotype. So as I mentioned, these are TALL, so they express cytoplasmic CD3 by definition, but they lack mature markers, and the original definition of this subgroup is negativity for CD1A and CD8, weak or absent expression of CD5, and then expression of at least one myeloid or stem cell marker, such as CD13, 33, etc., etc. And just if you look at that immunophenotype, it's not too dissimilar, in fact, from a lot of acute myeloid leukemias, or even the biphenotypic or mixed phenotype leukemias that we saw on the previous slide. So in terms of what we're doing at the moment, immunophenotyping is simple and provides obviously a practical classification system for allocating treatment, which is very important. And this has worked, and obviously we've had great successes in acute leukemia treatment, particularly in children. So it's a very good system, but maybe not ideal for everybody. So about 5%, up to 5% of acute leukemias, depending on age, cannot be classified using these systems. And as I mentioned, especially in the area of T versus myeloid, this can depend on a very small number of markers, CD3, MPO. This can be subjective, and really it can be difficult to distinguish sometimes between an immature AML, so that's M0 AML, versus an immature TALL, ETP subgroup, or even a mixed phenotype acute leukemia T myeloid. And the problem with this is that these cases often do poorly, so historically do very poorly. So the prognosis has improved for some of them, particularly the ETP subgroup, who now have a prognosis that's the same as other TALL subtypes in children at least. But they remain chemoresistant, and really the success is dependent on giving them extra treatment because they don't respond as well to the normal chemotherapeutic regimes. And really, these cases, it's likely that they arise from multi or oligopotent hematopoietic progenitors, or at the minimum, they share biological characteristics with them. So that's what we do at the moment. I just have one slide on the molecular landscape of this kind of general subgroup. And I don't want to go into this in detail today, but just to stress that these subgroups, so across the T myeloid spectrum, have very similar mutational profiles, particularly in cytokine receptor and tyrosine kinase signaling. So in TALLs, especially the IL-7 receptor JAK-STAT axis, in hematopoietic transcription factors, so things like RUNX1, which is common in ETP-ALL, mixed phenotype acute leukemia, and M0 AML, and also epigenetic molecules, so factors that affect the chemical status of either DNA or histones. So you can think of factors like PHF6, which is common in mixed phenotype leukemias, and also in TALL, a bit less common in AML, or core components of the polycomb repressive complex 2, like EZH2, CZ12, etc. So this suggests that there's maybe a common mechanism of oncogenesis in these leukemias, or maybe a common cell of origin. And it has been proposed that maybe this should drive treatment allocation a bit more specifically. So based on these common molecular abnormalities and potentially common molecular Achilles heels, let's say. So I haven't referenced it here, but there's a nice opinion piece on this a couple of years ago in the BJ Heen from Kensis and Gutierrez from Dana-Farber, who summarized a lot of the logic for this. So that's where we are. We have a good system. It doesn't work for everybody. These T myeloid cases, I think, are a difficulty, and we could do better. What I'd like to argue is that we're maybe in a position now to think about some of these difficulties a bit differently, and that this has been driven by the new technologies that we have in the genomic space in particular. So what we know about hematopoiesis today has, of course, been greatly helped by high throughput genomic approaches, which we all know have become more and more integrated into normal research use and now into clinical use, hopefully. These have told us about normal blood cell development, and with these new technologies, as often happens, this has driven the development of new approaches to analyze the data. So these enormous amounts of data, this has meant that computational tools to deal with them have had to be developed in parallel. And we wondered a while ago, could some of these novel techniques that are used in normal hematopoiesis, and particularly for single cell analysis, could they provide different insights into leukemia biology? So just to summarize some of these techniques, so I've classified these as traditional, in inverted commas, transcriptional analysis approaches. So I'm not sure how traditional they are, but let's say a little bit older. So hierarchical clustering, of course, has been around for a long time, but in terms of gene expression in cancer, this has been around for over 20 years, and this was a genomic first. So the first gene expression classifier used in cancer was in AML versus ALL, and this is back in 1999. And more recently, gene set enrichment analysis is another tool to compare basically enrichment for different transcriptional signatures between sample groups. So you take two groups and you essentially see which ones transcriptionally resembled a defined set of genes in kind of very simple terms. So this can be used to measure the activity of specific molecular programs or to assess the transcriptional resemblance to other cell types. So just to give some concrete examples of both of these, so I think hierarchical clustering of gene expression profiles has been shown in TALL to be very consistent. So there's a number of papers in the 2000s showing that when you take these gene expression profiles, you do unsupervised hierarchical clustering, you get this very reproducible separation of TALLs into these groups which are very much driven by the state of differentiation of the TALL. So for example, with these more mature clusters or these immature cluster here. And this gives you clues about the underlying molecular pathogenesis and allows you to identify sometimes important genes that are driving these cases. So also here, for example, these HOXA overexpressors. And then gene set enrichment analysis that I mentioned. So a couple of examples here. This is a paper from the Charles Mulligan's group in St. Jude's from 2012 on ETP-ALL. And they reported that although ETP-ALL was originally described due to its similarities to normal early thymic precursors, in fact, what they found was that these cases were more similar to hematopoietic stem cells and immature myeloid precursors than ETPs, so more of a kind of stem cell myeloid type leukemia. So this might give clues about the leukemia cell of origin. We've used this tool a couple of times. One example is this paper we wrote in 2017, looking at expression of ribosomal genes in CBF beta MYH11 positive AML, that's the M4EO subtype. And we found, so we had some interesting in vitro findings on the role of the oncoprotein in affecting ribosomal gene expression, so both ribosomal proteins and ribosomal RNA. And we were able to correlate that in vivo in independent patient groups, showing that these cases had a very strong expression of ribosomal genes that might play a role in the leukemia pathogenesis. So these tools are useful, but they do have some drawbacks. So they rely on comparisons of predefined groups. They're not really equipped to account for the transcriptional heterogeneity between individual leukemias, and they're also not equipped to resolve the relationships and understand why these groups come out differently. So there's newer approaches for doing this. So one example are these dimensionality reduction approaches like PCA or stochastic neighborhood embedding, so TISNE, which most of you will see being used for single cell RNA sequencing analysis. And briefly, these are tools which reduce this enormous multi-dimensional information you have in gene expression down to a couple of dimensions that are enough for a human brain to kind of process and look at, and essentially aid visualization of where these cases are in relation to each other, and look at the different relationships between these subgroups. Excuse me. So this has been used, these type of techniques have been used by different groups to look at biological differences between leukemias. And again, a paper from the St. Jude's group. This is a very nice paper in 2019 where they used stochastic neighbor embedding to help discover novel subgroups of BALL. And as it turned out, that was the differences in the position on these dimensionality reduction analysis was down to specific dysregulation of PAX5. And this group, they've had a couple of other reports of using this technique. For example, more recently at ASH, there was an abstract on using this to identify a subgroup of immature TALLs with BCL11B enhancer hijacking. So this approach is very interesting. And then we tried to take this look at this in a slightly different way. So I'm going to present a few slides on a recent work we did using these novel analytical techniques to look at differentiation arrest, and specifically the spectrum of differentiation arrest in leukemia. And this is work that has recently been published in Leukemia, has kind of been up online a bit before that. I have presented this in other fora, but I'm presenting some extra information today that I haven't shown before, which I think is an interesting angle on some of the techniques. And this is work that was done in my last job in the Hôpital Necker in Paris, led by Professor Elisabeth McIntyre, and in collaboration with Elisa Laurenti from Cambridge. And this is a very long-standing project where we did transcriptional profiling of a mixed cohort of acute leukemias, very heavily biased towards immature subgroups, and we used a variety of different techniques to try and work out the transcriptional kind of activities that were going on between different subgroups. So first thing we did, and which is really the root of the project, is that if you take these cases and do unsupervised hierarchical clustering, which I showed you already, is you see this separation whereby a subset of the TALLs cluster in the AML subgroup, suggesting that they were similar to AML cells. This held up when you confine the hierarchical clustering just to the TALL cases. And we also saw this on PCA, particularly in the first dimension. And you might expect this given what I've said already about the overlap between these cases, but this technique seemed to identify some different biological characteristics. So there was only partial overlap with other measures of immaturity in TALL, such as T cell receptor gene rearrangements, or ETP immunophenotype. So we went on to look in a bit more detail at what these AML-like TALLs were like. So just to remind you of the schema of hematopoiesis, and what we did was we took transcriptional signatures that had been already reported by Elisa Laurenti's group and other groups, and we used this gene set enrichment analysis tool to work out what the AML-like TALLs were similar to, let's say. And this is a summary of the analysis, and in fact, these cases were much more similar to immature myeloid progenitors, such as like monocyte progenitors or megakaryocyte erythroid progenitors, than lymphoid progenitors like this multi-lymphoid progenitor. So much more myeloid in their characteristics than lymphoid. And this was consistent in the PCA. We saw that these cases in blue kind of fell into the space around these immature myeloid progenitors. But interestingly, it wasn't just this. So this is probably the last proper bench work I did personally before I settled into a career of telling other people to do experiments. So this is work I did in the lab in Paris, where we had a very nice arrangement with the cardiothoracic surgery service. We received fresh neonatal thymuses that were removed during cardiac surgery. And I spent very long days dissecting these thymuses, depleting them, sorting them by facts. This is an example of the strategy, in particular for these immature populations. So these CD34 positive populations, and in particular these very immature CD34 positive, CD1A negative, CD7 negative populations, which you can see here. This is kind of a 8 o'clock in the morning till 10 o'clock at night job, and then at the end you end up with about 5 cells, and it's extremely frustrating. But eventually, with enough thymuses, I was able to get enough RNA to send for expression profiling. And of course, after all that, this ends up being a footnote in figure 1C, I think. But the interesting finding we had is that these AML-like TALLs, again, although they resemble these myeloid progenitors, they also are very similar transcriptionally to these rare CD34 positive, 1A negative, 7 negative populations that are in the thymus that, again, are known to retain myeloid potential. And this hasn't been reported before, that there are leukemias that are similar to this immature subgroup in the thymus. And if you remember, I said a couple of slides ago that ETP ALLs were actually much more like myeloid progenitors. They weren't like ETPs at all. So why is that? And we didn't focus too much on this in the paper, but it is on the supplementary data. And really, it's the point of this is just to stress that the GSEA results very much depend on the gene list. Okay, so this paper in Nature and our paper had very different sets of genes that were analyzed that were ETP genes. So the analysis was done by the same person, Elisa Laurenti, who was also on this Nature paper with Charles Mulligan's group. But the way this list was derived was by comparing the early thymic progenitors to bone marrow populations, essentially. So the differences in the genes that are expressed in early thymic progenitors basically relate to T cell genes, whereas in our analysis, we compared our early thymic progenitors with the later thymic population. So it brought out a different set of genes. As you can see, there's very little overlap between the gene lists and very different functional pathways when you look at this kind of GEO analysis. So just to stress, this is not to say that the previous analysis was wrong, but that the results of these GSEAs are extremely contingent, of course, on what you define your gene list to be. So just to be a bit circumspect on this, and I think our analysis would show that these immature cases certainly share transcriptional similarity with the earliest thymic progenitors. And then the other technique that we used, which we thought was the most interesting really, and we focused on in our publication, was this technique called ICGS, or iterative clustering and guide gene selection. And this is a technique that, like TISNI, has been mostly used to analyze single cell RNA sequencing data. And briefly, this relies on the identification of a small number of what's called guide genes that are enough to define coherent transcriptional modules. So basically, just by taking a few genes, you can define wider transcriptional programs. And this tool relies on serial clustering using these guide genes and refinement, serial iteration that goes through multiple steps. And then at the end, you get this separation, this clustering, and you can also do GSEA analysis at the end. And this is from the original paper that described it. And I think the nice thing about this is that they use the technique to analyze normal hematopoiesis. And I think what's nice is that it reflects these kind of dirty transitions you get between the different hematopoietic populations. So I've shown you kind of nice, neat figures where you have these populations around the place, but of course their identity is not, 100% one thing or the other. And I think the authors represented this quite nicely on this Waddington's landscape, where they showed that these discrete populations actually have activation of multiple different gene expression programs, so that as in many things, identity is quite complex. So we use this to analyze our cohort. So again, there's 124 cases, mostly immature, either TALLs or AMLs. The guide genes, as you can see, so these are identified in an unbiased fashion, and they were mostly hematopoietic genes, so myeloid genes or T lymphoid genes. You can see some examples here. Again, GSEA analysis correlates with different blood cell subtypes. And what you can see, I hope, is that what ICGS is able to do, so instead of having these kind of blocks of groups, You see this transition, so from the T lymphoid cases on the left, which have high expression of T lymphoid genes, which you can see here, to more myeloid cases on the right, with expression of myeloid genes. And then in between, you have these clusters, which we ended up calling interface acute leukemias, that have either very little expression of lineage discriminating genes or kind of a partial myeloid signature, let's say. And of course, all the TALLs are on the, or all this side is TALLs, all this side is AMLs, and then you have a mix of TALLs and AMLs in between. And this overlapped with our previous analysis by hierarchical clustering of these AML-like TALLs, so they were all in these interface groups as well. So again, we were interested in working out what kind of transcriptional programs are activated in these cells. So a similar type of analysis where we took these normal hematopoietic subpopulations, including our thymic subpopulations that I described. You see this lymphoid to myeloid transition again, I think. I hope you can appreciate where you get more myeloid type gene transcription in the myeloid cases, more lymphoid type gene transcription in the lymphoid cases. And then these interface clusters, which are a bit of a hodgepodge. So you have both activation of these lymphoid programs as well as these more myeloid gene expression programs. So this might be expected, but the thing that surprised us is that when we looked at what was driving this resemblance, it was actually the cases in the interface clusters that were originally diagnosed as AML that actually had more lymphoid-like gene expression. Okay, so just to restate that, we have a mix of gene expression in these interface clusters, but it's actually the AML cases in inverted commas that are more lymphoid than the lymphoid cases. So this was a surprise to us. We had some mutational data on kind of about 2/3 of the cohort with the targeted panel. Not to dwell too much on this, just to say that the mutational profile doesn't correlate with the classic pattern you'd expect in immature leukemias versus mature leukemias. PHF6 again was quite interesting in that we noticed there was a strong correlation with Notch mutation status. So in the presence of Notch mutation, these cases were in the more T lymphoid subgroup, whereas in the absence of Notch mutation, they were in the interface cases, suggesting that maybe, you know, addition of a Notch mutation does drive the cells more towards a TALL phenotype than AML. We used this technique on independent TALL cohorts with similar results, so focusing on TALL. And again, we could see the spectrum of differentiation, which I think is quite nice. That correlates with ETP immunophenotype in these cases. And also we have the similar patterns of gene expression GSCA analysis. So then we were interested in whether we could, you know, this is our cohort, but whether this was potentially clinically significant. It's difficult to look at in our cohort. It's very heterogeneous, and lots of different treatments, obviously. So tried to see whether we could find these interface-like cases elsewhere in the published literature. So what we did to do that was we focused on AML. We didn't really find too much going on in TALL. And we looked at 2 independent cohorts, and briefly what we did is we categorized cases as being interface high or interface low, i.e. having interface acute leukemia like gene expression, based on the similarity of their gene expression to our cases. So interface high and interface low cases, and this works at a variety of thresholds, but the kind of bottom line from the clinical point of view is that these interface AMLs, so we took 2 independent cohorts, a German cohort and a French cohort, and if you look at cases that have gene expression that's similar to our interface cases, they do very badly in terms of their overall survival and EFS, as you can see here. And we found this was independent of other variables and multivariate analysis. So it was quite strong, so independent of kind of classical cytogenetic predictors. Suggests that these cases maybe when treated on AML type treatment maybe don't do as well as they maybe might. The other thing that was intriguing about analysis of these independent cohorts is that similar to our group, that they also showed enrichment for lymphoid type gene transcription. So in both cohorts, the German and the French cohort, we found that there was this strong expression of a lymphoid, and particularly B lymphoid gene expression signature, which is kind of intriguing. And although these are preliminary data, the fact that these cases do badly on AML-directed treatment does make you wonder about other approaches. And we were interested in a paper from the Sobato group a couple of years ago, where they showed that RUNX1 mutant AMLs are responsive to steroids, which is of course a more lymphoid-directed treatment. And I know anecdotally there's been some encouraging results with treating AMLs with steroids from other groups. And this does correlate with what we saw in our cases, whereby all the cases with RUNX1 mutation were in these interface clusters. So preliminary kind of ideas, and which needs to be tested a lot more rigorously, of course. And I think I just have a couple of minutes to finish off with one other type of approach that I think is extremely interesting. So these are not our data. This is from another group. So again, to come back to single cell analyses, so these are really powerful techniques to kind of tease out not only the kind of bulk transcriptional differences between different leukemias, but to really look within the individual leukemias at the heterogeneity that's there, and really to have a highly granular analysis. And with the advances in the technology, not just the transcription in these cases, but also the epigenetics and the phenotypic relationships in normal cells and leukemia cells. And I just like to highlight this paper because I think it was really, really nice, which was published a couple of years ago using a combination of CiteSeq and AttackSeq. So very basically kind of at a single cell level, looking at the immunophenotype and the epigenetic chromatin accessibility in individual cells. And the authors of this paper used this to look at both normal progenitors and leukemias at a single cell level. And they used, again, a dimensionality reduction approach, in this case something called UMAP, which is similar to the other dimensionality reduction approaches, but there's kind of different underlying mathematical assumptions about the shape of the data. The shape of the manifold is the main difference. And they use this to construct these maps of normal progenitors, so the normal immunophenotypic and epigenetic landscape. And the nice thing about this, again, a bit like our data, is that, you know, it's not kind of discrete populations. These are continuums of transcriptional and epigenetic features. And they used these maps to map on mixed phenotype acute leukemias. And briefly, what they were able to do is look very specifically at differences in gene expression and transcriptional regulation in individual cells, which allowed them to interrogate very specific differences in transcriptional and epigenetic programs, and in this case allowed them to identify different regulation by RUNX1 of very specific gene expression programs that correlated with the poor prognosis. So that's just a whistle stop on that data, which I'd encourage you to look at, from Grandia et al. A couple of years ago in Nature Biotechnology. But again, an example of, you know, using these very powerful approaches to ask very, very pointed and specific questions about leukemia biology, or to reveal very, very specific features. So I think I'm just on time there. So just to summarize what I've shown you today. Really based around this idea of using the hematopoietic roadmap to think about leukemia. And I hope you'll agree that these newer analytical techniques that are available, and of course are coming on stream all the time, can provide novel biological insights, tell us more about the molecular pathogenesis of leukemia, and can inform potentially decisions about treatment and also the study of molecular pathogenesis. Obviously, now that we know more about the mutational landscape of leukemias, there's more focus on these epigenetic activities, nuclear architecture, and all different features, about many different features of the cell biology. And once again, it's very critical to relate this to studies of normal processes. So we're very interested going forward in our group in using these kind of techniques to understand leukemia better. Just to acknowledge, so the work I showed you on ICGS was done in my previous lab in Paris with our collaborators. I was supported by the Kate Kendall Leukemia Fund and Intermediate Fellowship during all that time, and I'm now an SBI, and these are our current funders at the moment. And this is, as Ava mentioned, I'm the Brendan McGonnell Professor, and this is Brendan McGonnell, whose legacy donation was responsible for creation of this post. So thanks very much to him as well. Okay, thank you.
Jonathan, thank you very much. It was a great talk. Jonathan, also first of all, to everybody, if you would like to have a question, you can type it into the chat box and I can read them out. But also there is a little reactions icon at the bottom of your screen, and you can raise your hand and you can ask your question as well. We have one question here from Philip Murphy, and Philip, you scooped my question here to Jonathan. And Philip's question is, have you looked at the role of the tumor microenvironment in gene transcription and epigenetics in these leukemias?
No, not at all. It's a very brief and boring answer to that, I'm afraid. No, we haven't. I mean, this is a very exciting area, and we were interested in this, Ava, and there's a lot of data coming out about this at the moment. It's not something we've looked at. But yeah.
So I was also thinking for these mixed phenotype leukemias, is it possible that there is hematopoietic stress ongoing in the bone marrow that pushes these cells to try to refill, the missing lymphocytic or myeloid population?
It's very possible. I mean, I think, there's a lot of evidence that, as you know, in terms of these immature leukemias that develop from clonal hematopoiesis, so particularly associated with DNMT3A mutations, that, it's maybe the stressed environment that drives that, or drives the clonal advantage that these leukemias have. So I think that's very possible. This increases with age. We know these type of immature leukemias and mixed phenotype increase with age, so particularly these M0 AMLs, ETP ALLs. So it's certainly a possibility. It's not something, again, we specifically looked at in our study so far.
Okay. Jonathan, I think we also work in similar studies and we do single cell transcriptomics, but I always feel that what we do is very far from the clinical practice. How do you see it, as a path that it could be implemented?
Yeah, absolutely. No, that's an excellent question. I mean, I think in terms of genomics in general, this is now part of clinical practice, I'd say, in most parts of the world. I think we're behind in Ireland, unfortunately, where there's a lot of people working hard to make that better. So I think in the first instance, getting that right is very important. I think you're right, single cell techniques will be very important, particularly in identifying kind of minor clones that are more likely to be chemo resistant, so earmarking cases where you really need to ramp the treatment. So this is a model that is already being followed in terms of correlation with mutational burden. So some groups rationalize treatment even based on, for example, very small quantities of mutations as measured by variant allele frequency. So if you have very small levels of a poor risk mutation like RAS, you still get intensified treatment. So I think doing that at a single cell level and maybe identifying cases that have odd cells that are very likely to relapse will be integral to treatment. I hope not in the not too distant future. As I say, we're a bit behind, but the rate of development of these techniques is obviously stratospheric. So maybe at BCNI Symposium 2031, we'll be talking about a routine use of this, hopefully.
Yeah, we will see. We will see, definitely. I don't see any other questions. So there are the final chance for people, if you would like to ask a question, either raise your hand or just start talking as well. That works too. You just unmute your microphone. And I think I'll just give one more chance to people if they would like to ask questions. I'll stop asking my own question. I could ask you probably another 10 of them.
Yeah, so, people are welcome to ask questions in the chat later.
Absolutely, absolutely as well. Feel free to use the chat box afterwards, even probably if you need some basic information. So we move on, and I would like to introduce our next speaker, who probably doesn't need any introduction, Owen Smith. So Owen is a really key figure in pediatric and adolescent cancer treatments in Ireland. He's professor of pediatric and adolescent medicine at University College Dublin. He's a consultant pediatric hematologist at the Crumlin Children's Hospital, and he's also the national clinical lead for child, adolescent, and young adult cancers at the NCCP, the National Cancer Control Program. Owen published over 350 papers and a real central figure in pediatric hematological and other cancers. With no further ado, Owen, I think the floor is yours to share your screen.
Thank you, Aoife, for that very nice introduction. So for something really different now from what Jonathan was talking about, and I'm going to concentrate on what we've been doing over the last number of years in trying to streamline children, but also adolescent young adult cancers into getting better treatment outcomes. And I'll end the talk by talking a little bit about a trial that we're about to embark on, so the so-called Altogether trial, which is a pan-European trial. The denominator is around 260 million, and we're hoping to actually recruit in the region of 7,000 to 8,000 patients into this trial. And the age is not going to go up to 16, it's going to go up to 44, with predominantly most of these patients will be under the age of 24 with acute lymphoblastic leukemia. So can I advance the slides, Eva? Don't seem to be able to do it from my end.
Is it not working for you, Owen?
No, oh, there we go.
Might have been just a bit delayed. Perfect.
Yeah, so just to remind people that the spectrum of cancers in children is totally different than we see in adults. And as you can see in the under 15s here on the left, the predominant cancer is hematological malignancies, leukemias, lymphomas, followed by central nervous system tumors. And then you've got a whole cadre of different types of tumors, from neuroblastoma in the very young to, you know, the sarcomas, the bone and soft tissue sarcomas in the younger adolescents. Now, most of these tumors are really fast growing and require intensive chemotherapy or intensive therapeutic approaches. But the good news is we cure in the region of 85 to 90% of these cancers. In relation to hematological malignancies, that is really exceeds 90%, with the non-Hodgkin lymphomas and the acute lymphoblastic leukemias around 95%. The unfortunate thing is there's a payback here, chronic complications of the drugs that we've been treating these patients with hitherto for, and we need to start addressing that. And another thing that's come out in the last number of years in relation to childhood cancers is the genetic predisposition. So in leukemia, it's around 1 in 10 of our families we would see there is a genetic predisposition driving the leukemia in children, whereas in the total group of cancers, it's around 1 in 4. So next slide. I can't seem to...
Owen, you need to just click on the actual PowerPoint presentation. You're in control.
All right. Oh, perfect. Thank you. So back in the early noughties, we started to centralize services for children in cancer, as everybody probably doesn't know, but a lot of the older folk in the audience will remember that we were treating especially leukemias in separately in Galway, in Cork, and in Dublin. And in Dublin, there were 2 different centres. But we decided then in the late 1990s, early 2000s, that we'd centralise these services into Crumlin. And as you can see, this is the sort of the demographic of what we do in Crumlin. We admit up to the age of 16. However, if you relapse within the first 2 years following your 16th birthday, we'll still treat you in Crumlin. And they're the numbers, so around 220 we would see every year of de novo and relapse diseases. The unit itself is 19 beds, there's 4 transplant beds, the whole unit is HEPA filtered, and a number of years ago we decided that we would build age-appropriate environment for 2 young adolescent beds. This was funded partially through the Teenage Cancer Trust in the UK, which I'll come back to later on. So the great thing now is that for the last 20 years or so, all diagnostic and treatment planning through specific MDTs are made through the National Children's Cancer Service at Crumlin. And the other important thing is that we share care with 15 units, pediatric units around the country. We're also with GPs, and in the last 10 years, parents. So we've actually taken parents on board as well to deliver chemotherapy in the home. Now, these parents are obviously vetted. They go through a competency program where they can deliver things like cytarabine to their child through an intravenous catheter in the home scenario, which really enhances their quality of life, etc. And this has been published in international peer review, led by Lorna Story and the group in Crumlin, and they've got a number of awards for this. So sharing care obviously is based around robust evidence-based supportive care protocols, and these were developed over the last 10, 15 years. They work incredibly well. We're very actively involved in academic and phase 3 clinical trials, as we'll come to in a few minutes. However, we've also recognized that we have a paucity of phase 1, phase 2 investigator-led trials, and we're trying to address that in the next cancer clinical trials HRB call. A number of us, obviously, on the clinical side as well, are PIs and co-PIs on international trial consortia. So centralization does have a number of positive impacts, and this was a review done by Charles Stiller and the group in Oxford, published in 2015, looking at cancer mortality across the European Union, comparing it to what was being done in the UK. And I'm pleased to say, as you can see here, that in green, out of 31 countries assessed during the period that we were using the UCAL 2003, Ireland came out on top in terms of overall survival. So this is a direct reflection of centralizing, a service to get better outcomes. Now, just a little diversion here. So when we talk about adolescent, young adult cancers, it gets a bit confusing in terms of young adulthood. It's not so much with the definition of a child or a definition of an adolescent, as you can see here, up to 0 to 14, 15 to 19, etc. And for young adolescents or young adults, then it's slightly different, and it's slightly different across the European Union, as you can see here, as I put down on the bottom of the slide. In Ireland, like in the UK, we've adopted that young adulthood goes up to 24. And we wanted to keep that specifically in mind when we were coming to constructing the so-called National Cancer Policy that came out in 2017. So this is a special or a specific cancer community, the AYA community. As you know, cancer is the leading disease-related cause of death in this group, and there are unique tumors that appear in this particular group, and the incidence is increasing, and specifically increasing in terms of lymphoid malignancies. So it will have an impact on what we do in hematology in the coming years. This slide here just shows you the different tumor types, and as you can see in the under 15s, the predominant malignancy is hematological, up to 40%, followed by brain tumors and then neuroblastoma and the sarcomas. And as you go into the AYA grouping, you can see that it falls off, the hematological malignancies fall off, but not that greatly in the under 20s, I would say. It's around 35%. And in fact, I will say as well that ALL is the commonest cancer in the under 21 years of age, which is something I don't think is fully appreciated in the hematological or in the cancer community. So it's still a big number of patients that develop ALL in the under 21s. As you can see also during adolescents, young adults, tumors like thyroid tumors, melanomas, germ cell tumors and sarcomas start to increase in this particular group. Now, this particular group also have unique needs. Fertility preservation is becoming more and more prominent in that adolescent young adults know that there's a great chance of surviving, and they really want to, preserve their fertility. And we get this all the time in the younger adolescents in Crumlin. Survivorship is an issue, transition is an issue that needs to be addressed, and psychosocial well-being is a huge area of concern for this particular group of patients. And something that I didn't really appreciate until I went to train in London, especially at UCL and what was called the Middlesex Hospital at the time, that age-appropriate environment is really high on their agenda in terms of where they're treated. This was a very nice paper published in the UK back in 2011. in the Journal of Adolescent Young Adult Oncology by the group from UCL. The title was Your Place or Mine, and they did a very nice survey across the UK in terms of AYA and where they wanted to be treated. And as you can see, they wanted to be treated in their own space, a place where, you know, they want to be treated like adults, but have all the privileges of children. They want a place not just to go to bed, as is the case in a lot of the units around the country and across the European Union. So age-appropriate environments leads on to better psychosocial well-being, etc., etc. So these are real significant areas of concern for this group. The British, as I say, really started to develop this whole area back in the 1990s when I was training in London. And as you can see, there's over 32 now. This is 28, I suppose, but 32 teenage adolescent units, young adult units across the United Kingdom. As you can see, the little file there for Dublin that we developed, a 2-bedded one in Crumlin, and we'll come back to that later on in the talk. And then limited survival improvements for specific cancer types have been highlighted for this particular group over the last number of years. And poor clinical trial enrollment is really one of the main reasons for this. And as you can see, this is some NCI data. And as you can see in the under 14s or the children, there's over 50% of patients are entered into peer review, usually randomized phase 3 clinical trials. And then when you get into adolescence and young adulthood, these patients fall off a cliff. Basically, they're not being entered into clinical trials. And there's lots of reasons for that, but there's also lots of implications for this as well. It's responsible for poor improving survival. And also it's, you know, it prevents them from getting novel therapeutic interventions as well, because normally these are brought in phase 2, phase 3 clinical trials. And the biology of a lot of these tumors is very ill understood. And the reason for that is if you're in a clinical trial, you tend to have tissue tends to be biobanked and then it tends to be interrogated, as Jonathan was saying earlier on, with specific new technologies. So, I mean, I suppose Philadelphia like ALL is the classic example here, where it was discovered basically by Monique Boer and Charles Mulligan's group in St. Jude. Monique was working at Rotterdam at the time. She's now in Utrecht. But they showed in a classic childhood ALL population that there was this particular entity called Philadelphia-like. And then when that was pushed out into the adolescent, younger adult population, this is probably as high as 1 in 4 of patients in the adolescent young adult population has Philadelphia-like ALL. Also, poor AYA representation in other studies as well, supportive care, quality of care, cancer epidemiology. So we really need to start thinking about getting these patients into clinical trials. And hence, back in 2016, I was asked with a number of other people. John Kennedy chaired the strategy group, and I was asked to specifically comment on children and adolescents and how do we bring this group forward. And I'm pleased to say that this particular strategy recognized, A, that Crumlin was the 9th designated cancer center, but also the adolescent cancer community was unique and their needs, or clinical unmet needs, were going to be addressed in the policy. This is one of the recommendations by the policy, that the HSC would ensure age-appropriate facility is designated for adolescents and young adults with cancer, specifically in relation to the new Children's Hospital that's based, as you all know, in St. James's. And this is just a couple of drone photographs from last month showing that this particular facility now is out of the ground. It's on a 12-acre site. The St. James's campus is a 40-acre site, and it's going to have 390 inpatient beds. It's the floor plate is 160,000 square meters, and this particular facility is going to be handed over to Children's Health Ireland in December of 2023. So from 2024, we're going to be able to move into this facility. And what does it mean for children and adolescents? Well, we're on the first floor of the inpatient facility that's mapped out here in green, both the inpatient and the outpatient. Our inpatient capacity is going to increase by approximately 50%. As I was saying to you earlier on, 19 beds now becomes 28 beds. We're going to have more adolescent beds. And there's going to be age and psychosocially appropriate infrastructure or inpatient facilities, including inpatient and outpatient den facilities. That this is one of the one of the things that adolescent young adults really want to have when they come into hospital and have their treatments. And I suppose one of the big changes here is we'll be treating pediatric cancer centres that have clinically medical driven conditions. As you know, we have the highest rate of Down syndrome in the European Union, and Down syndrome has a 30-fold increased risk of developing a hematological malignancy. So our age limit is going to go up to around 20 years, as you can see on this particular slide. And so what does that mean in terms of numbers? This is data from 2015 to 2017. I got this from the Cancer Intelligence Group within the NCCP. And as you can see, we're dealing with roughly around the same numbers between 16 and 24 as we do between 0 and 15. And these are the pediatric-centric cancers, the leukemias, the lymphomas, central nervous system, malignant bone tumors, soft tissue sarcomas. And as you can see, there's a significant number of these within that age group of 16 up to 20. And what does this do? This translates into what we'll be seeing is around 40 to 50 more patients into this facility when we open it in 2024. And then for the older patients, or the older AYA patients, 20 up to 24, as you can see, there'll be somewhere in the region of 150 of these patients that we will be thinking about putting into specific adolescent young adult centres that we are developing. And you'll hear more about that in a second. So the cancer strategy then, recommendation 27 was that the HSC will develop closer links on a hub and spoke model between the National Cancer and Child and Adolescent Centre at CHI and the other designated cancer centres around the country, and more specifically a commitment to appointing adolescent young adult oncologists, hemato-oncologists, and all of the other ancillary stuff for age-appropriate psychosocial care like social workers, psychologists, palliative care, etc. And so in March 2019, the NCC appointed me as the clinical lead, as Elisabeth said earlier on, and we had our first meeting in April 2019. And the membership is right across the board, from pediatric medical oncology at all levels, from medicine, nursing, allied healthcare professionals, but also we were very keen on bringing in patients and parents, the PPI component to this, and also the non-governmental organizations as outlined below here. And we developed five main subgroups, as you can see here: fertility preservation, shared care, palliative care, survivorship, and communications. And then within the first service planning, we got approval for AYA clinical, an AYA cancer doctor, an AYA assistant director of nursing, and then now in post, and we're about to appoint an MDT coordinator at grade 7 level. And then just briefly then, the hub and spoke model is we've been involved from the NCCP point of view, looking at the 8 designated cancer centres and asking them what actually do they have in place in their particular cancer programs that relate to AYA. And we're going to select in the first instance 4 of the designated cancer centres to have a designated AYA program, and then the NCCP and myself will promote sort of further developments of an AYA program within these particular cancer centres. The MDT is not your typical MDT that you would see in adult or indeed in children. It's going to address all of these AYA specific needs, which I won't go into in any detail. And it's also going to address the thorny issue of clinical trial enrollment, availability, and participation. And that brings me on to this particular slide that, you know, most of you are awaiting the outcome of the HRB decisions on the cancer cluster applications for funding for the next 5 years. We put in an application from Children's with UCD, their clinical research program, headed up by Peter Dorn, Cormac Owens, and myself. And we put in a very comprehensive application to fund not just children's, but also up to 24. So we're going to try and capture all children, young and older adolescents, as well as young adults up to the age of 24 into clinical trials. These are the strategic objectives that we laid out in the application. And some of them there that are emboldened we're specifically interested in, especially around relapse patients, getting them onto phase 1, phase 2 trials, but also leveraging the excellence that's been done in developing shared care across the pediatric program. Can we do that with the AYA hub and spoke program, but also with the group in systems biology, Walter Kolch, and indeed Jonathan's group in UCD, and Mark Lawler in Queen's University, to develop an all-island pediatric as well as an AYA HEMONC trials and research program. And again, as Jonathan said, we're quite behind in terms of genomics and cancer, and this is an area we've highlighted in the application as well. And we're very keen that every child, not just with leukemia, but with cancer, are sequenced. And the reasons for that is not just to understand the basic biology of the leukemia, especially at relapse, but also to look at the not just the somatic, but the germline genome as well, and specifically in relation to genetic predisposition to toxicity, because it's still a huge burden for our patients. If you look at survivors of 30 and 40 years, on average around 20% of those patients will have significant chronic disease. So it's an area that we're up for, and we are in negotiations that everybody will, with childhood and adolescent, young adult cancers, will have a pipeline to whole genome and RNA sequencing. And then also within the strategic aim is to network across Europe as well as across Ireland, and I'll come to the Altogether Consortium in a few minutes, and also to increase our entry into clinical trials, especially in phase 1, phase 2 trials that are open to the older patient. We'd like to consider children for those as well, but also the older patients, and the exemplar here is the Altogether trial of the older patients coming into basically a pediatric protocol going forward. And this is all in keeping with a pan-European strategic objective. This is a paper I wrote with a number of people across the European Union that came out in ESMO Open last month, or this month, I can't remember, last month or this month, just mapping out what is needed across the European Union so we can have a fairly straight level playing pitch for all AYA patients in terms of their diagnostics and their treatment planning. So again, it's in keeping with European policy, and this has been fully endorsed by the European Union. So when we come to ALL then, as I was saying, in 2002, everything was centralized into Crumlin. There were a number of groups using UCAL-based protocols, ourselves at National Children's Hospital, Cork with Michael Madden, Ernest Egan across in Galway. Crumlin at the time were using a different type of protocol, but we decided when we amalgamated and we all moved in together that we would continue on with UCAL protocol, so every child would be entered into a phase 2 or a phase 3 clinical trial. And I was involved in a lot of this stuff going back into the 80s at Great Ormond Street with Ian Han and Judith Chessels, and before me, Ian Temperley was involved. He was the national coordinator. And as you can see, survival over the decades really has increased. And our last trial, it was a very successful trial, the 2003 trial that ended in 2011, recruited almost 3,000 patients, and the overall survival was in excess of 90%. And what fell out of that particular trial was that MRD assessment after induction was really very telling in terms of overall prognosis. As you can see here, if you've got a low risk or negative MRD after induction, you have around a 95% chance of being cured at 5 years. Intermediate risk 86, and high risk around 80%. Now, during this trial as well, word got out that basically that adolescents, the older adolescents or the younger adults, if they're treated on pediatric protocols, because in the 1990s, a lot of work had come out of COG, a lot of work come out of the US and across Europe as well, that if this patient group between 16 and 21 were treated on pediatric protocols, then their overall survival actually jumped by around 25–30%. And then in UCAL 2003, during its time, we started to recruit more adolescents, as you can see, to 16 plus years of age. And what fell out of that particular trial, interestingly, was that the event-free survival was around 75%, which was around the 25% difference from these patients being treated on a more adult orientated trial. And when you looked at MRD for this particular group as well, it was very interesting that if you had a negative MRD at the end of induction, your event-free and overall survival was in excess of 95%. Again, showing that the power of molecular monitoring was really significant in getting more of these patients treated, but also the adolescents now are really, or the older adolescent, young adults are really benefiting from this pediatric-centric type protocol. And just to summarize, the UCAL 2003 was the first randomized trial to show stratification of treatment by MRD response improves outcome. We could get rid of the second delayed intensification if you had a very good MRD signal. And those who had a bad MRD signal, they did benefit by increasing the amount of chemotherapy or intensifying their chemotherapy in an augmented approach. So this led beautifully then on to the Altogether Consortium, which basically started to come together back in 2012, 2013. And it's a conglomeration across the European Union, 14 countries. The denominator is around 260 million. And as I was saying earlier on, we're hoping to recruit during the lifetime of this particular protocol around 8,000 patients. The preconditions and the hypothesis around the protocol, as I was saying already, failures of current ALL treatment is due both to under and over treatment. We overtreat some patients who don't really need it, and we lose some of those patients from toxicity. And we undertreat some of these patients that in the past we've deemed to have standard risk, but hidden behind that standard risk is a number of molecular determinants of relapse. And also further general intensification is unlikely to improve outcome. We think we have really come to an asymptote in terms of intensifying treatment for this population, and we do believe, therefore, we can, look at alternative therapies that are less toxic, like antibody-based and cellular-based immunotherapeutic approaches for this particular protocol, and also the whole area of, can we integrate genetic and molecular treatment response in a better way than we did in the previous trial? And Anthony Mormon, who works out of Newcastle, has done an awful lot of work over the last number of years in pulling the data set together. That's going to be the basis of how we stratify these patients going forward. This is the genetic stratification. I'm not going to go into this in any great detail, but just to highlight the high-risk side of genetics, the good risk side of genetics, and we're going to address the Philadelphia-like, or at least the Ava class fusions within the Philadelphia-like cohort. And then also we're going to marry in, copy number alterations in terms of gene array, and we can come back to that in the chat if you wish. So we're going to pull that all together and integrate that with thresholds of MRD response. And again, I won't go through this in any great detail, but each individual sort of, or each subgroup of genetic classifications will have a different threshold for MRD. So this is all going to be integrated into a computer model when all of this data is produced during the trial. And this basically is not to be taken down, but this is basically the roadmap as in terms of how we risk stratify these patients. So we start off with diagnosis. This is, and then, if your standard risk, you reach a 3-drug induction, and then if you're high risk, you read a 4-drug induction, you have your MRD, and then you are sorted into either standard risk, intermediate high risk, and then the intermediate risk is then further subdivided around 50-50 into intermediate low, intermediate high, based on genomics and MRD response. And this is the elevator summary of that, breaking it down into standard, intermediate risk, high, low, high risk, etc. And just to highlight that Down syndrome ALL is going to be treated separately. It's a separate protocol within the Altogether. And the reason for this is, as we all know, Down syndrome ALL, we treat probably per capita the greatest number of Down syndrome patients within the European Union. And a lot of the work that's been done by our group in Crumlin have really shown that these patients' supportive care is a major issue in treating Down syndrome. And we believe that we can get rid of some of the toxicities from the drugs by substituting some of these drugs with immunotherapy like blinatumomab, which we'll see in a second. And then can we use more experimental therapy, less toxic, et cetera, et cetera. And as you all know, blinatumomab and CAR T-cell was passed by, or got approved by the FDA and the EMA back in 2017. So that's what we're going to be doing in this particular protocol, introducing antibody-based and cellular immunotherapeutics on the backbone of the altogether. And that backbone essentially is based on the UCAL 2003, our protocol from back in the early noughties. So we're going to introduce blinatumomab, CAR T-cell, and then some of the patients are going to get rid of some of the chemotherapy, the patients who are standard risk, who don't need a lot of this therapy. So this is the diagnostic approach using clinical and genetic and molecular treatment responses during induction and consolidation, and then going on to the master protocol. And then patients will be pulled out to have de-intensification in a randomized fashion in those patients with standard risk or intermediate risk low. Patients then in the intermediate risk high, will be another randomization using inotuzumab, but also an experimental optimization of maintenance therapy. As you all know, we use 6-mercaptopurine predominantly as the thiopurine in maintenance, and we're going to be integrating thioguanine into this protocol as well, because there's a lot of evidence, especially from the Danish group, that you get better maintenance control of ALL when you introduce both of these drugs into maintenance. And then obviously the ones who've got Abel class fusions, they will be started on imatinib. They will be intermediate risk high, and then patients who are deemed to be high risk, they will be pulled out and then will be offered CAR T-cell through the Cassiopeia protocol. And as I was saying earlier on, the patients with Down syndrome will be on a separate protocol, will be risk stratified again by MRD, but will be given blinatumomab, 2 cycles of blinatumomab, trying to reduce again the toxicity burden for these patients, but also increasing the efficacy signal going forward as well. So these are the interventions, 3 randomizations, de-intensification, which is music to my ears, because we still do lose a lot of patients who shouldn't, not from their disease, but from toxicity. And then how do we increase survival or increase cure, leukemia cure, by introducing antibody-based and cellular-based immunotherapeutics, as well as targeted therapy with TKI for the Abel class fusions. So this consortium then, as I was saying to you, over 14 countries. One of the last, well, this is the last meeting in person that we had. We held that in Farmleigh back in November 2019. This was the penultimate meeting before the sign off of the protocol. There's a number of people here that are present at this particular forum today. They can look at themselves there. And the important thing about this particular consortium, it's been built to last. In other words, it's been built to move on. This is going to be phase 1 or part 1 of this particular consortium, which is the ALL01 protocol. We've had our first AGM earlier on this month, and there's a number of people from Crumlin involved, and indeed Larry Bacon is involved in this as well from St. James's, but predominantly Crumlin people. There are 3 of us on the board. Jonathan is on the board, I'm on the board, Andrea Malone's on the board, but also I'm on the product study group, as well as Andrea Malone, and Jonathan heads up, chairs the Biobank Committee. John and myself are on the Science Committee, etc., etc. Amelie from Crumlin is also on a number of these groups. So it's really a very comprehensive group across the European Union, and I think for something like ALL, this is the way forward, and hopefully this model as well will be adopted across other groups, other cancer groups in the adolescent young adult sphere. So hopefully I've convinced you in the early part of the trial that treating adolescent young adults with cancer, especially ALL, is very different from treating cancer in ALL and young adults. So this is an area we have been behind in the past in terms of how we treat them, where we treat them, but also, you know, the molecular determinants of these diseases now are going to really guide us in how to reduce the burden of toxicity in this population and also get more of these patients cured going forward. So I think that's my last slide, and I thank you for your attention. And this is the new Children's Hospital where adolescents as well as young children will be treated from 2024. Thanks very much.
Dear Owen, thank you very much. Excellent. That was a very interesting talk and a great update on what is happening. I think we are all waiting for the new hospital to really open. Philip has a question in the chat, and I can start with that one. I'll just read it out. Thank you, Owen, for a fantastic and inspiring talk. I would be especially interested if you're in your thoughts on fertility preservation in young women.
Yeah, great question. I mean, it's really something that's been a huge issue. So what we've been doing, Philip, with the Marion Fertility Clinic in Holles Street, we got a grant funded by a grant from the Irish Cancer Society, 400,000 euros. And what's happening at the moment is we're doing a pilot study with them in relation to exactly the population you're talking about. So you'll be hearing more about this. At the moment, we're doing a feasibility pilot study, and hopefully in the next 6 months, we will have a system up and running for this particular population. Yeah, as you know, we don't have, we've got oocyte preservation facilities. It's ad hoc, and it's across the private sector as opposed to be publicly funded. The Harry unit was the one that did it in the past, but we really need to prove in this pilot study that this should be funded by public purse and available to all people through the HSC. Thanks for that, Philip. Yeah, very important point.
Very much. Owen, you mentioned that there are MRD tests throughout the Altogether trial. Do you think it can be introduced into clinical practice on a regular basis?
Yeah, so we do it all the time now. So for this particular trial, it's you know, depending on what lab you're using across the European Union, the MRC or the UCAL trials use a PCR-based assay, and so, but you don't need a PCR-based assay. It can be a flow-based assay, and for this particular trial, we will be hoping to do all of that work within Crumlin for the group. Yeah.
Very good. We have one more question from Mary Cahill. Thank you, Owen and Jonathan. In view of the many connections between TLL, ALL, and the myeloid-type features of some ALLs, can you please comment on these leukemia and the future therapies? Should we have acute leak protocols rather than ALL protocols?
Right, Jonathan, do you want to go? Do you want to take that?
Yeah, I can. Yeah, thanks very much, Mary. Yeah, I think that's something I was partially trying to highlight during the talk. So these aren't new ideas. I mentioned there's a very nice piece by Kensis and Gutierrez a couple of years ago in the BJ Heme suggesting that there should be an entity of AMTL, so acute myeloid T leukemia, based on the shared molecular susceptibilities. I think we're moving that way. Obviously, at the moment, for practical reasons, we're very much constrained by the structure of trials and all the practical things we have to deal with. But as we move towards this kind of famous notion of personalized medicine, I think I would hope that in the future we could have a much more directed approach to the molecular landscape of the leukemia rather than just the phenotype. The other suggestion is whether, I've been asked in these type of conferences if that kind of differentiation happens a bit later. So you start off and you just basically have one acute leukemia regime, as you allude to, and I think that's a very interesting point. I think I'd refer more to the active clinical people about that, whether that might be a bit toxic to offer to everybody, for example, first off, would be my feeling from a distance. But yeah, I think that's a really interesting idea.
Yeah, I agree. Yeah, great question, Mary. It's a bit like what Jonathan's talk was about. It's a bit like infant ALL as well. it has mixtures of myeloid as well as lymphoid sort of biology. And we did a randomization in the previous trial looking to see would a myeloid approach be better than a lymphoid approach. And the answer is it comes out no, but it sheds some insights into MRD response to these patients as well. You know, for the TALLs going forward in this particular trial, the full protocol is yet to be signed off for the TALL patients, but as you know, these are usually high-risk patients, and the whole area of using, say, something like daratumumab is being discussed in a randomized way for this group. But again, MRD really drives this as well, the good actors versus the bad actors. So it's a really interesting space, TALL, at the moment. There's a lot new happening in it. A lot of the biology is starting to be unfolded, unlike, you know, it's like B-cell precursor back 20 years ago. So I think we're going to see a huge amount of advance in TALL going forward. Yeah, thanks, Mary.
Thank you very much, Owen.
Just
We move on.
that, Owen, just to say it's historically neglected as well. I mean, we've got TALL in with BALL, of course, but it's very different.
Yeah, sure.
Thanks, Mary.
Thank you very much. We move on to our next speaker, Dr. Andrea Malone. Andrea is going to talk about pediatric relapse refractory AML and the European phase 1 study on inotuzumab. Just a brief introduction. Andrea received her BSc in medicine and in surgery from UCD, and then specialized in medicine 2 years ago. And she's also working at the Crumlin Hospital. So welcome, Andrea, to the meeting.
Okay, thank you, Eva. Can everybody see my slides?
Yep.
Yes, we can.
Great. Okay, hotspotting is working in Crumlin.
Thank
We're
you.
in a black hole. Mobile phones still work. So thanks for the opportunity to present this study. So this is a study that we in Crumlin have been involved in over the last number of years. Just to give a brief background on acute lymphoblastic leukemia, as everybody knows, it's the most common malignancy in childhood. And as Owen has showed in his slides there, it's actually curable in greater than 85% of cases now with multi-agent frontline protocols. The need in leukemia remains in the relapse disease, where it still portends a very poor prognosis, and there is an urgent need for new therapeutic approaches. This is just a graph showing again, as Owen did earlier, the progress that has been made in the treatment of de novo ALL over the last number of decades. And then on the right, you can see that in relapse disease, only about half of the patients are salvaged with high-dose chemotherapy and generally stem cell transplantation. So the focus of this study was on inotuzumab, which is an antibody drug conjugate and consists of a humanized monoclonal antibody against the cell surface adhesion molecule CD22, which is expressed in 90% of B-cell precursor ALL in children. It's conjugated to a potent anti-tumor agent, calicheamycin, which is internalized into the cell, induces double-stranded DNA breaks within the nucleus, and thereby causing cell death. The inotuzumab has been extensively studied in adults and with very encouraging results. And one of the seminal papers was in the New England Journal of Medicine in 2016, where inotuzumab was tried against standard of care for relapsed refractory ALL in adults. And it showed significantly higher CR rates in the inotuzumab group with higher MRD negativity rates, and it concluded that it was better than standard of care. This led to the appetite for exploring this drug in pediatric relapsed refractory ALL. And we already had in vitro data showing that sensitivity of childhood ALL cells to calicheamycin, and some limited existing data from a phase 2 study of inotuzumab in relapsed refractory leukemia that included 5 pediatric patients, along with data from compassionate use programs in Europe and the States, totaling 63 patients. So this study was undertaken by the Innovative Therapies for Children with Cancer, or ITCC consortium in Europe, of which CHI is a member, and it is a phase 1 multi-center single-arm open-label study of inotuzumab in CD22 positive relapsed refractory B-cell precursor ALL, and the findings of this study were published in Blood earlier this year. So the design of the study, it included children greater than or 1 but less than 18 years, and they were recruited over a 2.5-year period. They had relapsed or refractory ALL that expressed CD22, and the primary objective of the study was to determine the maximum tolerated dose and recommended phase 2 dose. Secondary objectives were to establish or to investigate the safety of the drug, response rate, as well as pharmacokinetic and pharmacodynamic data. A modified rolling 6 escalation design was used, with de-escalation based on dose-limiting toxicities during the first course of treatment. Inotuzumab dosing was assigned at study entry, with dose level 1 being 80% of the established adult dose, which was 1.4 milligrams per meter squared. This was given in a fractionated dosing, one course being 3 doses of inotuzumab given at day 1, 8, and 15. Okay, sorry, I'm just not able to advance my slide there. Okay, patient characteristics. So there were 23 evaluable patients. All of them were over a year. Most of them were actually over 6 years. 60% of them were in second or greater relapse, and the vast majority of patients had an M3 marrow or greater than 25% blasts. They had a median of 2 prior therapies before receiving inotuzumab, and over half of these patients had had a previous bone marrow transplant. So the results of the study were very positive, with an overall response rate of 80%. Of these responders, 84% of these were MRD negative, and the median duration of response was 8 months, being non-significantly longer at dose level 2, with 3 prolonged continuous complete remission at 15 and 19 months following inotuzumab therapy. This amounted to an overall survival of 63% at 6 months and 40% at 12 months. And of note, the median survival after dose level 2 was not reached. No further escalations were made to dose level 3, given the excellent results and the expected limited further improvements that would be made at dose level 3, and also given that dose level 2 was the established adult dose. Toxicity was mainly cytopenias, and all patients had at least 1 grade 3 adverse event, usually thrombocytopenia, anemia, and neutropenia. Notably, there were no cases of sinusoidal obstruction syndrome. This graph A on the left shows that at dose level 2, the blue block shows that the vast majority of patients received an MRD negative remission. To summarize, inotuzumab was very well tolerated in children. It had notable efficacy in heavily pretreated children with relapsed or refractory ALL that expressed CD22. The recommended phase 2 dose was established at 1.8 milligrams per meter squared, as in adults, given as fractionated dosing on day 1, 8, and 15. There was a high overall response rate of 80%, with a non-significant superiority at dose level 2. There were high levels of MRD negative remissions and a longer duration of response at dose level 2. As regards the pharmacokinetic and pharmacodynamic data of inotuzumab, this was comparable between pediatric and adult doses, and adult patients. And inotuzumab is now being trialled in combination with reinduction chemotherapy in relapsed refractory ALL in children. So just like to acknowledge the ITCC consortium. This study was sponsored by Erasmus MC and the hematology team and clinical trials unit in Crumlin, along with our patients and families. Thank you.
Andrea, thank you very much. We have one question here from Mairead. Thanks, Andrea. Did you expect less VOD in the children, or were you surprised by no cases?
Yeah, so there were no cases of VOD in the children who received inotuzumab. 2 patients did subsequently develop VOD, and those 2 patients had got high dose chemotherapy. They weren't actually transplant patients. So it was encouraging that there was no VOD. I know in adults, the rate of VOD can be is a concern, especially if these patients go on to transplant.
Thank you very much. There's another question. Is there data on combining inotuzumab with blinatumab in relapsed childhood AML?
I'm not aware of data on combination. I know in our patients here, in relapsed refractory patients, we have used it sequentially. Obviously, inotuzumab is better for bulky disease, and we have certainly used blinatumab following inotuzumab as a bridge to transplant.
Thank you very much, Andrea. Are there any other questions? I didn't see more in the chat.
Yeah, there's one from Ezat there, who says, can prior therapy impact on CD22 expression, Andrea, and subsequent drug sensitivity? So that can certainly happen in adults.
Yeah, well, obviously all of the patients had to be CD22 expressing. They were all heavily pre-treated, relapsed refractory patients. So it certainly didn't affect the patients in this study. And we find in our relapsed refractory patients that they almost all still express CD22 despite previous multi-agent chemotherapy.
Andrea, thank you very much. And we move on to the last speaker of the first session. Andrea, would you mind sharing your screen? Thank you very much. Our final speaker of the morning session is Peter McCarthy, and he's going to talk about searching for targetable gene fusions in childhood leukemia. Peter is a clinical researcher, a clinical research fellow in the Jonathan Bonds group, and he's a the systems biology island researcher. Peter, you're very quick sharing your screen.
Apologies, sorry.
No, you're excellent. You're excellent. I looked down to my paper and the next thing I saw your first slide up. Thank you very much. The floor is yours.
Thank you very much. Good morning, everyone. And thank you for the opportunity to present this morning. My name is Peter McCarthy. I'm a hematology specialist registrar and clinical research fellow based in the National Children's Cancer Centre in Children's Health Ireland at Crumlin and Systems Biology Ireland in UCD. In my talk this morning, I hope to highlight two recent examples that we have come across in patients of ours in Crumlin, and the beneficial use of targeted therapy to improve outcomes and reduce toxicity in children with acute lymphoblastic leukemia. As has previously been mentioned by the other speakers, the most common childhood cancer is acute lymphoblastic leukemia, and there's two main subtypes, B and T. This image just shows an example of some of the current targeted therapies that are in use in patients at present for the various different subtypes and the different targets. There's a number of these different agents, for example, the use of tyrosine kinase inhibitors in Ph positive or BCR-ABL1 positive ALL. A more recent example, and one which was referenced already, is the example of the use of targeted therapy in Ph-like or BCR-ABL1-like ALL. And in these conditions, basically they have a gene expression profile resembling that seen in BCR-ABL1 positive patients, but in patients lacking the fusion itself. There are a number of recurring activating kinase alterations that are seen in Ph-like ALL, as you can see from this image, and these affect multiple pathways. For this talk today, we'll be focusing specifically on the ABL class fusions, that's ABL1 and ABL2, and the use of tyrosine kinase inhibitors specifically targeting these. The first case I will discuss today is that of a 6-year-old female who presented with a very high white cell count, greater than 500 by 10 to the 9 per liter, and a large mediastinal mass. That is a large swelling in the center of the patient's chest, compressing their airway and the surrounding lung tissue. She also presented with a significant complication of the high white cell count, known as tumor lysis syndrome. She underwent a diagnostic workup and was ultimately diagnosed with T acute lymphoblastic leukemia. At the time, FISH revealed a TLX3 rearrangement. The patient would have been managed as per the UCAL 2009 interim guidelines in Crumlin Hospital, initially on regimen B due to her high white cell count. Then at the end of induction, due to a high-risk MRD, she was switched to regimen C and proceeded through regimen C. until she underwent a routine full blood count prior to commencing her maintenance therapy, which unfortunately revealed a relapse of her disease at this time. She underwent a repeat workup for her condition, and cytogenetics at the time of her relapse revealed the presence of a NUP214 ABL1 rearrangement, in addition to some other abnormalities. There is an example here of a close-up of this patient's microarray, which is used to detect expression of thousands of genes at the same time and can show gains and deletions of genes of interest. NUP214 ABL1 is also of interest in that it is a result of episomal amplification. And this means that there is an extra chromosomal genetic element that has the ability to exist autonomously, freely replicate in the cytoplasm, and then be reintegrated with its chromosome and then replicate with it. We wanted to look and see whether the NUP214 ABL1 rearrangement was present at the patient's diagnosis or only at relapse. We obtained samples from diagnosis, remission, and relapse, and we performed a targeted PCR followed by Sanger sequencing. And we were only able to demonstrate the presence of the NUP214 ABL1 rearrangement at the time of the relapse. NUP214 ABL1 is sensitive to ABL1 kinase inhibitors, and previously have been shown to inhibit the proliferation of leukemic cells expressing this protein. This allowed us to integrate targeted therapy with the TKI dasatinib for this patient, who ultimately proceeded to a hematopoietic stem cell transplant and remains in remission post her transplant, and is currently completing 2 years of dasatinib maintenance therapy. Our second case is that of a 10-year-old male. He initially presented at 5 years of age, again with a high white cell count of greater than 200, was diagnosed with TALL with a TLX3 rearrangement. He proceeded to be treated as part of the UCAL 2011 clinical trial and completed therapy on regimen B. Unfortunately, 5 years after his initial presentation, the patient relapsed. Again, a diagnostic workup would have been performed at the time of his relapse. He had a microarray performed, and as mentioned previously, the microarray shows various gains and losses in the patient's leukemia cells. I would draw your attention specifically to the abnormalities highlighted in chromosome 1 and chromosome 12 on the patient's relapse sample. Gains are evidenced in the blue lines above the central line, and losses are evidenced in the red lines below the central line. We hypothesized that this may have been due to a translocation of ETV6 ABL2, which would have been due to the clonal evolution from the patient's original disease. We aimed to perform FISH analysis to ascertain whether this was present at the patient's initial diagnosis or only at the time of his relapse. The FISH in this case was certainly much more challenging, as the breakpoints are unknown for ETV6 ABL2. We were able to perform a multiplex PCR followed by a more targeted PCR, which was able to followed by confirmation with Sanger sequencing, which was able to confirm the presence of this rearrangement at the patient's diagnosis and relapse. The ETV6 ABL2 protein is sensitive to ABL1 kinase inhibitors. And this is another example of the use of targeted therapy being integrated into the patient's management, again with the use of the dasatinib TKI. The patient ultimately proceeded to a hematopoietic stem cell transplant and also thankfully remains in remission and continues on TKI maintenance therapy for 2 years following his transplant. Thank you very much for the opportunity to present today, and I hope my presentation also offers you some insight to the use of the targeted therapies in the clinical realm, and hopefully how we can, as we begin to expand the diagnostic opportunities that we have for these patients in terms of whole genome sequencing, that we can integrate more targeted therapies in patient management going forward. Thank you very much.
Yeah, Peter, thanks a lot. These are really interesting 2 cases, and it really showed the power of genomic sequencing and identification of chromosomal aberrations. You went through very quickly on your second case study slides where you mentioned clonal evolution. Did you detect all the initial aberrations at time of relapse?
So we did, but I suppose with to be fair, I should have mentioned in the slide, so the microarray that we would use at the time of the patient's relapse, that technology wasn't available to us at the time of the patient's initial diagnosis. So we would have repeated the patient's diagnostic samples with that technology at the time of relapse. So we had more information, I suppose, at the relapse than at the time of diagnosis initially.
Okay, so it's definitely a nice case of clonal evolution where you had all the original aberrations and new ones.
Mm-hmm.
And
Mm-hmm.
I assume it would happen in the same cells. I know that your analysis couldn't reveal this.
We presume that is probably the case. I suppose that our analysis didn't reveal that as such, but the fact that the rearrangement was present in the patient's diagnostic sample would make you consider that would be the possibility. But we didn't prove that for certain.
Perfect. There are also two questions came in through the chat. One is from Larry Bacon. Thanks, Peter. How close are we to having these molecular tests as part of our routine diagnostics?
A really good point. So Professor Smith already alluded to the Altogether trial that is going to be opening in Ireland, hopefully later on this year. And one, I think, strength of an international trial like Altogether is that it's going to really integrate these kind of diagnostic tests into the patient pathway. And we're going to be specifically looking for PH-like mutations in these patients, initially with targeted FISH analysis, and then hopefully in time, as patients start to get more access to next generation sequencing through that approach.
Okay. Thank you very much. And Mary Cahill has another question. Thanks, Peter. Did the group have any diversity open on the best TKIs to use?
So that would have been discussed as to whether imatinib or dasatinib would be preferable. I think there's less information in the literature about this. A lot of this would be based on case reports, and it seems like dasatinib has been shown to be quite effective in these patients, and that was discussed at the kind of the MDT, and that was felt the best approach to take.
Okay. If there are no more questions, no, I don't see any more coming in. Peter, thank you very much. And this concludes our first session. I'd like to ask the speakers of the second session to stay online with us, like keep your cameras on. And we ran a little bit over time, so we are going to reconvene at 11:05. Okay, I hope to see you all back in about 15 minutes time.
So I'm Professor Mary Cowell. I'm one of the BCNI members, and I'd like to thank Ava and Sharon, who put in all the hard work to this conference now, in fairness. And we did consider whether we should actually postpone it in view of the cyber hacking. But then we thought, look, we'll just go ahead, and whoever can come, can come. And we're very gratified at the number of you that are making the effort, because we know, especially if you're anyone in the HSE today, has a lot of other calls on their time, and everything is taking longer than it should. So without more ado, it's my great pleasure to introduce Dr. Richard Dillon, who's a clinical senior lecturer in cancer genetics in the Department of Medical and Molecular Genetics in King's College. So I believe he works in the School of Basic and Medical Biosciences, which is part of the university there, obviously. And he's a consultant in Guy's Hospital as well in London. So he's got a special interest in AML and in the genetics and molecular genetics of that, and has been in touch with us about a number of patients here in the last number of years. So I'd like to thank him on a personal note for that, and to welcome him to give us his presentation this morning. Thank you.
Thanks so much for having me. I'm Richard Dillon from Guy's Hospital and King's College London. My talk's going to be about some molecularly guided interventions to prevent relapse in AML. So first of all, what I'll do is I'll just briefly review the current cytogenetic and molecular risk stratification frameworks that we're using for AML, particularly to decide which patients should be transplanted. And then I'm going to go through about how we can use molecular MRD assessment to refine outcome prediction for patients within each of these groups, and how we can use that to refine their treatment to try and prevent relapse. So first of all, in the favourable...
No.
Are you hearing me okay?
Yes, we can. Probably someone didn't have the microphone muted.
Yes, could everybody mute their microphones, please? Or if we can do that centrally, that's good too. All right, Richard, my apologies. Please carry on.
But I'm coming through okay, am I?
You most certainly are, yes.
Great.
I think the issue
Thank
was
you.
you had a little competition there, but it's
Oh,
sorted
don't
now.
worry. And then secondly, in the patients with that remain in the intermediate risk group, this is the group really where decision making, particularly about first remission transplants, has historically been the most difficult and challenging. And I'm going to show how we can use MRD to provide additional information that can help to inform those very finely balanced decisions for each patient. And then finally, I'm going to talk about patients who are in the high risk group, where possible, these patients should all receive an upfront transplant where possible. But however, we can still use molecular MRD techniques in these patients in order to optimize their pre and post-transplant management. And I'll go through some real life examples of each of these cases. And then finally, the final point is in all of the risk groups, we can use sequential measurements of molecular MRD status for early detection and preemptive treatment of relapse. And I'll go through some potential options that we can use in this situation, and again, using some real life cases. So first of all, I'd just like to briefly review the current risk stratification system for AML. And as you will know, the most powerful prognostic information is still provided by cytogenetics. And this slide is a very famous slide that was produced by David Grimwade, looking at almost 6,000 patients who were enrolled in a series of the MRC AML trials. And what you can really see is that each cytogenetic abnormality really has its own prognosis, to the extent that we're almost dealing with a whole series of different diseases. And of course, there are a large number of these. And in order to make this clinically useful, David proposed this simple 3-group risk stratification system, which is still in use, and it's known as the MRC prognostic classification. So we have a small number of abnormalities that are considered favourable risk, APL, and core binding factor leukemias. We have a selection of abnormalities that are considered high risk, and then all other patients are classified into the intermediate risk group. Now, of course, this still leaves a significant proportion of patients in the intermediate risk group, and this makes management decisions for these patients more difficult. And over the last 10 to 20 years, there have been a huge amount of efforts to further refine risk stratification within this intermediate risk group using molecular techniques. And the first was really the demonstration by Rosemary Gale and others that using a mutational status of just 2 genes, we can stratify a number of the patients in this intermediate risk group with a normal carrier type into patients that do exceptionally well or exceptionally badly. So the patients that do exceptionally well are patients without a FLT3 ITD mutation with an NPM1 mutation, and the patients that do exceptionally badly are patients with a FLT3 ITD who are wild type unmutated for NPM1. And of course, when we overlay that onto the existing cytogenetic risk classification groups, what you can see is those patients who are in the favourable molecular risk group have equivalent outcome to patients with favourable risk cytogenetics, and patients with adverse molecular risk have equivalent outcome to patients with adverse risk cytogenetics. So this is quite an advance. However, this still does leave a significant number of patients into this intermediate risk group where it becomes much more difficult to know how to treat them. And further progress on these patients was really made with the advent of next generation sequencing. And when the first next generation sequencing studies of AML performed, it became very clear that AML is extremely heterogeneous on a molecular level. So this is a study by Tim Lay and colleagues who did whole genome sequencing or whole exome sequencing on 200 cases of AML. And they found that each case had, on average, they had 13 mutations. And they found 200 genes that were mutated in 2 or more cases in this cohort. So this is a exceptionally complex molecular landscape. And why this is relevant is that a number of these mutations have prognostic impacts. So for example, P53 and ASXL1 in addition to RUNX1 mutations. What you can also see from this figure, which is from Eli Papimanuel's paper looking at the German AMLSG cohort, is that there is an interaction actually between the mutational and cytogenetic status. So on the left-hand panel, you can see that there's an interaction where patients with TP53 mutation and complex karyotype have a much worse outcome than either alone. And patients with ASXL1 mutation have significantly worse outcome in the presence of a co-occurring SRSF2 mutation. So there's a whole extra level of complexity that's been revealed by these next generation sequencing studies. And in order to try and distill this information into a clinically usable format, this has led to the development of the ELN, European Leukemia Network Risk Classification Scheme, which attempts to integrate results from the cytogenetic and molecular profiling incorporating next generation sequencing studies. So what you can see is in the favourable risk group, as well as those patients with core binding factor favourable cytogenetics, we also include patients with NPM1 mutation without FLT3-ITD or with a FLT3-ITD with low allelic ratio. And into the adverse risk group, we include those patients with this NPM1 wild type FLT3-ITD high allelic ratio genotype, as well as patients with mutations in RUNX1, ASX1 and TP53. And as a broad rule of thumb, the way this is intended to be used is that patients in the favourable risk group would not normally be considered for transplant in first remission. Patients in the adverse risk group should be considered for a transplant in first remission at all possible. And in the intermediate risk group, we have this decision is much more finely balanced. We have to decide this based on MRD and other patient and donor-related factors. So what I'd like to point out is, although the ELN risk stratification is clearly very powerful, actually when you look within the ELN risk groups, there's still a degree of heterogeneity. So for example, even in patients with favourable risk groups, actually almost 60% of them will die from their disease. Sorry, only 60% of them will survive and the remaining 40% will die from the disease. And in the adverse risk groups, there are a small number of patients who become long-term survivors. So we can start to use molecular MRD techniques to try and refine risk stratification even within these ELN-defined risk groups. In the favourable risk group, this can be then used to identify a small number of patients that have a very poor molecular response who are predicted to have relapse and intensify their treatment. And in the adverse risk group, we can use the MRD to try and optimize their therapy, both pre and post-transplant. So I'd like to go through each of these three groups in turn and just point out a few particular considerations. First of all, considering patients with favourable risk or core binding factor translocation AML, which is inversion 16 and T821. Now, of course, these patients have an ideal MRD target, which is the translocation breakpoint, which represents a sequence that is not normally found in healthy individuals. It's entirely leukemia specific and can be amplified by qPCR methodologies with very high sensitivity. However, it's been well documented for these patients that they may have ongoing expression of the fusion transcript at very low levels for years after completion of treatment. And this does not necessarily predict relapse, perhaps because the translocation occurs in a pre-leukemic clone, which requires additional molecular hits to develop full-blown leukemia. Fortunately, in the NCRI AML15 study, John Yin and colleagues were able to define a series of thresholds of translocation copy numbers normalized to housekeeping gene, below which relapse is extremely unlikely. So you can see on the left-hand panel, this is for patients with T821 leukemias. Below the thresholds defined by John Yin and colleagues, patients have only a 7% risk of relapse. This is the majority of patients. Whereas if patients are persistently above these thresholds, relapse is almost inevitable. And similarly, on the right-hand panel, you can see this for patients with AML with inversion 16. And these thresholds have broadly been validated now by other cooperative groups, particularly the German group. So we can really use this very practically in terms of monitoring patients at the end of treatment and identifying which patients are in this group, which is at a very high risk of relapse, and select these patients for early intensification of therapy in order to prevent relapse from happening. I'll move now to talk about patients into the intermediate risk cytogenetic group. And of course, the most common intermediate risk cytogenetic abnormality is a normal karyotype. And roughly 50 to 60% of these patients will have a mutation in nucleophosmin. This also provides an ideal target for MRD monitoring using quantitative PCR for the reasons that, first of all, it is an insertion mutation, typically 4 base pairs in length, in a predictable part of the gene, in actually the tail end of exon 12. And this allows a patient-specific primer set to be used to specifically amplify the mutant allele with no amplification, little or no amplification of the wild type allele. And the other thing is NPM1 mutated AML have very high expression of the mutant transcripts, and this allows us to have very high sensitivity in the range of 1 in 10 to the 5 to 1 in 10 to the 6 in optimal samples. Adam Ivey and David Grimwade showed in the AML 17 cohort, which was the trial just prior to the current AML 19 study, looking at patients with NPM1 mutated AML and looking at their MRD status in the peripheral blood after the second course of chemotherapy. They could readily divide patients into those who had a very poor outcome, who were MRD positive in the peripheral blood at that point, with a 3-year overall survival of only 24%. And this contrasted dramatically with patients who were MRD negative in the peripheral blood at this time point, who had a 73% 3-year overall survival. And this led to the recommendation in the subsequent AML 19 study, which is still ongoing, currently recruiting, that those patients who are MRD positive in the peripheral blood should have their treatment intensified. So those patients were allowed to go into the high risk arm, which is the randomization between CPX 351 and FLAG-IDA, and they were recommended to receive a upfront allograft. And what you can see is the early results from the AML 19 study on the right-hand panel. This is just looking at those patients who are MRD positive in the peripheral blood post-course 2. And you can see that their outcome seems to have improved quite significantly. And their 3-year overall survival, excuse me, is currently sitting at around 68%. So we hope that will continue to be the case with prolonged follow-up and further recruitment. So for this group, early intensification does seem to be having a difference. However, there are also a number of other cytogenetic abnormalities in the intermediate risk group, which could also be molecular MRD targets. The most common of these is perhaps the T911 MLL AF9 fusion. Now, for these patients, the kind of data banks that we're using in order to inform our clinical decisions are much, much more limited, because the MLL assays have not been internationally validated in the same way. But however, we can still use information from these assays to try and improve personalization of clinical decision making for these patients. And I'll show you an example from our local MDM in which this information was very useful. So this case is a 44-year-old female who had therapy-related AML by virtue of the fact that she'd had a previous breast cancer and had been treated with chemotherapy and radiotherapy, which included anthracyclines. She had a reduced cardiac ejection fraction, and she had poor donor options. Her karyotype showed a T911 MLL AF9 fusion. She had no mutations in FLT3, NPM1, or actually any other genes on our standard gene panel. So we're able to track her disease using the MLL AF9 fusion transcripts, and the expression level of these transcripts at diagnosis afforded a sensitivity of 1 in 10 to the 5 to 1 in 10 to the 6. And what you can see here is plotted the MRD level, normalized to the diagnostic sample, which is given a level of 1, and plotted on a logarithmic scale. And what you can see here is the post-course 1 sample, in which the bone marrow is plotted in red and the peripheral blood is plotted in blue. And both of these samples tested negative at very good sensitivity. And the post-course 2 bone marrow likewise showed a molecular complete remission at good sensitivity. So bearing in mind these results, and the patient's specific situation in terms of a cardiac dysfunction and poor donor status, we decided this patient should not receive a first remission transplant, should have consolidation chemotherapy and very careful monitoring, saving the transplant options in case there was any evidence of molecular failure. And I'm pleased to say that to date, she has remained in molecular complete remission, remains very alive and well. And then the second application of molecular MRD monitoring, apart from risk stratifying patients, is to use this in a sequential manner in order to for early detection and then hopefully preemptive intervention to prevent frank relapse in patients with molecular failure. And I'll show you an example, another real-life example of that here. And this is a patient that was enrolled into the current AML-19 study. This patient was a woman in her mid-50s who had no prior medical history, presented with AML with NPM1 mutation and a FLT3 TKD or tyrosine kinase domain mutation. She entered AML-19 and had a standard induction, but unfortunately had primary refractory disease, had intensification with FLAG-IDA and had a bone marrow transplant, which you can see on the green dotted line here. And due to various post-transplant complications, she didn't end up having MRD reassessment actually until day 189 post-transplant. And the results of that again are shown here. And with the blood on the blue line and the bone marrow on the red line. So one thing I'd like to point out about these plots, which is very important if you're going to be using these in clinical practice, where a sample is negative, we plot that result at the maximum sensitivity that would be afforded by that particular sample. And this can be affected by a number of factors, the most important of which is the quality of the RNA, which is in turn is impacted on by factors like the transit time and the quality of the sample. And you can see that this is plotted at just a 2 log from diagnosis, meaning that the sample only afforded a sensitivity of 1 in 100. So this is really suboptimal for MRD point of view. We should be able to actually get, we should be able to detect MRD down to the level of 10 to the 5 to 10 to the 6 in a good sample. So we advise early repeat, and this was positive. At about just above a 2 log reduction from diagnosis. We never take any action based on a single sample because these assays can be vulnerable to a number of technical artifacts such as false positive, sample switches, contamination, etc. So we requested an immediate repeat sample, and you can see that the level has gone up by 1 log. And this therefore fulfills the European Leukemia Network definition for a molecular relapse, which requires 2 consecutive samples to be positive with an increase in transcript levels. So this patient will definitely relapse and relapse very soon without any intervention. Of note, she had quite severe graft-versus-host disease at this point, and this affected her skin, mouth, and external genitalia. So what are the options that we could consider for the treatment of molecular relapse in this patient in order to prevent a relapse? As you know, in the post-transplant setting, frank relapse would be associated with a very poor outcome, so we'd like to do everything possible in order to prevent this from happening. So the first option many people would think of is a donor lymphocyte infusion. Of course, this would be contraindicated in this patient because of the active graft-versus-host disease. However, where it's possible to deliver donor lymphocyte infusions, the outcomes are relatively favourable. This is data from the AML-17 study. And what you can see on the blue line is the overall survival from the date of molecular relapse for those patients who received donor lymphocyte infusion for molecular relapse. And their 3-year overall survival was 79%. And you can see on the red line that this is patients who did not receive donor lymphocyte infusion. This could have been for a number of reasons, including early progression. So this is clearly not a randomized comparison, but makes the point that donor lymphocyte infusion can be effective when patients are able to receive that. This patient was not able to receive donor lymphocyte infusion, so we then have to consider other options for treatment of molecular relapse. So the first of which may be a consideration of venetoclax-based treatment, particularly in view of the fact that this patient has an NPM1 mutation. This figure is from a case series that we published in the British Journal of Haematology last year. This is for 12 patients who experienced molecular relapse who were treated with venetoclax and either isocytidine or low-dose cytarabine. And the patients with the blue lines, these are patients who regained molecular complete remission. And as you can see, this was regained in 11 of the 12 patients. One of the 12 patients, unfortunately, only had a very transient molecular negativity and then relapsed very quickly. Importantly, this is the only patient in the case series that had a FLT3 ITD mutation. And ITD mutations appear to, there's preliminary evidence suggesting that they are providing an escape mechanism for venetoclax-based therapy. Which brings us on to the option of using FLT3 inhibition for this patient. As you would remember, I said that the patient had a FLT3 tyrosine kinase domain mutation at diagnosis. Now, of course, before we start to use FLT3 inhibition, we'd really like to know, is the FLT3 mutation still there? So for this patient, we applied a technique called droplet digital PCR, and this technique allows identification of low-level point mutations. What happens actually is the PCR reaction is partitioned into an emulsion of tens to hundreds of thousands of oil droplets, each of which acts as its own endpoint PCR, and these are then run through a machine that's very similar to a flow cytometer and classed as either being positive or negative. And this really is a good way to eliminate background, which you in a traditional endpoint PCR for a point mutation you would typically see background, which prevent you from calling mutations at a low level of less than, say, 5%. So in this patient, it's actually a multiplex reaction. The FLT3 wild type amplification is shown by the green dots, and patients with FLT3 wild type plus the D835Y mutation, which this patient had, is shown in the brown dots. And you can see that was positive at a level of just under 1%, consistent with the molecular status. So with that information in mind, we went on to give the patient a gilteritinib FLT3 inhibitor, and you can see that this results in a reduction, and then in MRD, and then molecular complete remission. Finally, I'd just like to briefly talk about the group of patients with high risk AML according to the ELN. So the first point to make is a number of these patients have balanced translocations, which are again ideal molecular MRD targets, and those are shown on the red asterisks at the top. And then these patients, shown in the purple asterisks, may also have methodologies that can be applied to monitor their disease. This genotype, which I mentioned earlier, which is the high molecular risk genotype, NPM wild type, FLT3 ITD high, I'll come on to talk about this shortly. Why would it be valuable to have a molecular marker to track these patients' disease? Because we know they're all going for transplant, but however, there are a number of things that it could influence. So for example, it could influence how many cycles of induction treatment you give prior to transplant, and how intense that is, whether they should just have D8 or whether they should have FLAG-IDA, etc. Secondly, in the peri-transplant period, we could use this to inform withdrawal of immunosuppression and administration of DLI. And finally, as mentioned before, we can apply this for early detection, preemptive treatment of relapse. I've got a couple of real-life cases to show you. So this is a patient from the My Child study, which I'm aware is also running in Ireland. I'm not sure if there's any paediatricians on the call. This patient, as karyotype is shown here, which actually did not suggest the presence of an MLL fusion, but in the My Child study, all of these patients are having extended FISH panel and RNA sequencing performed, and we're finding a surprising number of particularly MLL fusions that are cytogenetically cryptic in this cohort. And in this patient, we found MLL AF10 fusion. So this patient received study protocol, mitoxantrone, Mylotarg, FLAG-IDA, and a transplant. And you can see the MRD profile here, and what's shown is the MLL AF10 fusion transcripts normalized to the diagnostic sample on a log scale as before. And what you can really see is with the induction and consolidation chemotherapy, really is only a very suboptimal reduction in the transcript burden. And actually, on the day 30 bone marrow post-transplant is still positive. Now, as I mentioned, we don't have a huge amount of data banks for these MLL patients, but the one published case series in this setting showed that all patients with MLL fusions who had MRD positive post-transplant all relapsed. And indeed, so therefore we strongly advised this patient to be monitored very closely post-transplant. The day 60 marrow shows the molecular progression, which is confirmed on a subsequent sample. Amazingly, this patient, despite the high transcript levels, is still in morphological remission. And this patient then went on to have treatment with venetoclax azacitidine and then a lymphocyte infusion. And this resulted in molecular complete remission. However, consistent with emerging data for the use of venetoclax azacitidine and MLL fusions, what we're seeing is they do respond, but the response tends to be very transient, as indeed once the treatment was stopped, the patient rapidly progressed and is now receiving treatment with an experimental drug, menin MLL inhibitor, to which at the moment, such wood, they're still responding. And then I just quickly like to talk about this high molecular risk genotype group with FLT3 ITD without NPM1 mutation. So what we're doing in the current AML19 study is we're screening all of these patients for cryptic fusions So we're screening them for NUP98 NSD1 and we're screening them for DECCAN because these can be cytogenetically cryptic and are strongly associated with FLT3 ITD. And we find that we detect these in approximately 20% of patients. Why is it important to know that? Well, first of all, on the left-hand panel, what you can see is that these patients do much worse than average. They have very poor survival, and therefore it may be important to try and optimize their treatment. And the second thing, of course, that these fusions result in an ideal sensitive specific molecular MRD target. And I'll show you an example of how that can be useful. So this is a patient in AML19 who is a 44-year-old female with de novo AML who had this high molecular risk genotype. She was randomized to FLAG-IDA in ML19, and meanwhile we screened her, as is routine, for the cryptic fusions, and she was positive for NUP98 NSD1. So then we were able to feed the MRD information back to the clinicians by the end of the second cycle of treatment. And what you can see is that she had an adequate MRD response to the first and second cycles, but after the third cycle, which she was randomized on the high risk arm to CPX351, there's been molecular progression on therapy, which is a very bad sign. The levels are back to the levels at diagnosis, but remarkably, there was no excess of blast by morphology. The patient therefore went on to have a transplant. However, we advised them to do very early post-transplant bone marrow, and the day 30 bone marrow is again positive, and the repeat shows molecular progression. This patient was then started on quizartinib and received rapid withdrawal of immunosuppression, and that resulted in a molecular complete response. And my final case is another patient from the MyChild study. This is another patient with this high molecular risk genotype. So in this particular case, we did the screening for the cryptic fusions, DECCAN NUP98, and this was negative. However, as I mentioned in the MyChild study, we're performing RNA sequencing on all patients to try and define molecular markers in as many children as possible. And RNA sequencing in this particular case revealed a cytogenetic cryptic fusion in NUP98 SP140. And the beauty of the RNA sequencing technique is really that it allows us to see the fusion junction sequence at the exact base pair level, allowing us to design a patient-specific assay for each child. And we were able to use this information to feed back to the paediatricians in real time after actually the first course of treatment. And you can see that they had a very poor response to the first two courses of treatment, but remarkably became negative after the transplant. However, bearing in mind the very kind of high risk early profile, the clinicians decided to do very careful post-transplant monitoring and found the day 60 marrow was positive. Fortunately, bearing in mind the poor early kinetics, they'd already set things up to receive, for the patient to be able to receive sorafenib, and they'd also harvested DLIs for the patient. And they were able to administer both of these in combination to regain disease control. So that's the end of my talk. And just to summarize, I've talked about how we can use molecular MRD in all of the ELM risk groups. In the favorable risk group, we're looking to identify that small proportion of patients that are high risk of relapse for early intensification of treatment. In the intermediate risk group, we're really looking to provide additional data to inform finely balanced and personalised management decisions, particularly regarding transplant. In the high risk group, we're really looking to be able to provide information that will allow clinicians to optimise pre and post-transplant management. I talked about some different options that we can consider for molecular relapse, such as donor lymphocyte infusion, venetoclax and FLT3 inhibition. Finally, I briefly touched on how novel diagnostic techniques such as digital PCR and RNA sequencing could be applied to patients in real time to inform and optimize the management and to try and prevent overt relapse from occurring. Thank you so much for your attention. Thank you so much for inviting me to this meeting. I really appreciate it.
Great, and thank you, Richard, for a super talk. It's so lovely to have you as well. So I'm just going to get straight down to the questions, if you don't mind, because you're going to be busy. Amjad from Galway wanted to know if you could comment on the KIT abnormalities in favourable risk AML and what impact they might have. There's quite a few other questions as well. So back to you.
Yes, so the KIT, KIT mutations do increase the risk of relapse in core binding factor leukemias. However, there's a paper from the French group, Jourdan et al. In Blood, showing that when they have the MRD information, actually the MRD information outweighs the KIT status. So for those patients who are KIT mutated but MRD below the relevant thresholds, actually they don't appear to be at increased risk of relapse.
Okay, thanks for that. nice summary. Trina Nicanila wants to know, she says, lovely talk, any mechanisms associated with the venetoclax and 5-ASA resistance? What do you think about that?
What I think about that is it's still a bit of an unknown. There's a really nice paper in Blood last year, Ingsu Tiong, Courtney Dinardo, looking at all of the combining all of the phase 2 data from venetoclax and looking at how FLT3 could provide a resistance mechanism. It's not completely clear. What appears to happen is actually they respond, but the response is transient and that there's a rapid relapse. I still think that's not really worked out, I think. That's going to be a focus of future work over the next few years.
Focus for future work, that's fair enough. Nina Orfali is complimenting you on your talk and says, do you have a scope in the near future for MRD monitoring of MLL, ALL with fusion partners apart from AF9?
Yeah, actually, so in the AML19 study and now in the MyChart study, every patient with an MLL fusion, we've managed to design an assay for them, even with kind of completely novel MLL fusions. And we've done that using next generation sequencing techniques like the RNA-seq that I've showed you. That's actually kind of routine in our lab now. So if you have any cases with MLL that you're struggling with, you know, we're more than happy to also run them on that. And actually, when you have a non-standard MLL fusion, the treatment decision making is even more difficult. So, you know, with 9/11, it's intermediate risk and 10/11, it's high risk. But if you've got a really kind of novel partner, you don't know what risk they are. So actually the MRD can be even more useful potentially in those patients.
Okay, thank you. More compliments from Ezat El Hassadi, who wants to know, in a patient with high risk with AML with TP53 disruption, do you see clonal evolution with resistant clone post-chemo? And if so, do you think chemo-free such as venetoclax is better in this cohort? So it's about clonal evolution and avoiding chemo, going to ASA and venetoclax potentially.
It's a really good question. I mean, they do so badly with chemotherapy, and you wonder if they might do just as well with venetoclax as a bridge to transplant. They do quite badly with that treatment as well, from what we've seen. And there was a paper from Andrew Way in Blood just this week showing that what they've described is that patients do have a transient response to venetoclax, but then there's some kind of evolution, but it's not a kind of classical clonal evolution as such. In some way, some of the cells manage to develop some kind of resistance through non-mutational means, perhaps epigenetic mechanisms or similar, and then they then cause disease progression. So it's not what you would call clonal evolution in the classical sense, but it is a form of that. So I don't know if you saw the ASH abstract from MD Anderson looking at their whole series of P53 mutated patients. Actually, when they looked at patients with either more than one P53 mutation or a VAF of more than 40%, they didn't have any long-term survivors at all, and they were treated with a mixture of induction chemo and venetoclax azacitidine. So none of the treatments are really good for these patients, is the bottom line. Unfortunately, and this is an area where we hope there will be advances, such as, you know, these anti-CD47 agents may be possibly starting to improve the prognosis for these patients in the future. At the moment, the treatment remains really suboptimal, and I think this point, sorry, there's a big boat going past.
London traffic, eh?
Whether the venetoclax-based or the chemo-based approaches, that would be a very good subject for a clinical trial, actually, I think.
Okay, and one final question. Rory Hargan wants to know, do you have any updates on the early stage of the Victor study?
Oh, thank you for knowing
Maybe
about
that's
it.
a bit of a cheeky question, but...
No, we're opening this study next week. We're hoping to.
Okay.
So the Victor study I guess it follows on from the last question. It's a randomization between intensive chemotherapy and venetoclax for NPM1 mutated patients without the FLT3 ITD who are 60 plus. And the endpoint is molecular failure. So patients will be able to switch over if the arms, if it's a crossover design, if there's molecular failure.
So that note to end, just like to thank you again for your talk and pass on the appreciation of all the audience. Thank you.
Thanks so much for having me.
You're so welcome. So it's now my pleasure to move to the next talk, and thank you to Richard for releasing his screen there. And I'd like to introduce Dr. Bruno Paiva, who is a research fellow in the Department of Hematology and Immunology in the University of Navarra in Pamplona. And he's the director of the flow cytometry core in the University of Navarra. Dr. Paiva's main area is in multidimensional flow in hematological malignancies, and he spans the myeloid lymphoid spectrum because he's interested both in MGOS and also in AML. So without more ado, I'd like to welcome Dr. Paiva, thank him for coming, and we'll be looking forward to your talk.
Thank you, Dr. Cale, for the very kind introduction, and also to the Blood Cancer Network of Ireland for the invitation to participate in the 2021 symposium and to talk about disease monitoring in multiple myeloma. I'll try to be brief, 25, maximum 30 minutes, and leave 5 or more minutes for discussion. I think this is clearly a relevant field in AML, and it was really a pleasure to see before the presentation from Dr. Dillon, but also in myeloma. And I truly hope that this slide, which is illustrative of data coming from the more recent clinical trials, is also reflecting what is happening in Ireland and in other countries, which is the fact that approximately half of patients, perhaps those that are younger transplant candidates, may achieve a negative immunofixation with current optimal treatment. And we should be aware that among patients with a negative immunofixation, many other laboratory tests are not informative. This would be the case of morphology or even immunohistochemistry because normal plasma cells predominant over abnormal within the plasma cell compartment, and therefore simple kappa lambda ratios will not be informative. And nowadays, with highly effective regimens, the percentage of tumor cells that remain after treatment in a patient in complete remission is approximately 0.001%. Not only from a cytological point of view, but also in terms of free light chain ratio, while its value is unquestionable across the spectrum of monoclonal gammopathies. Truth is that there is now a great body of evidence showing that in patients in complete remission, the serum free light chain ratio is not informative, and therefore the stringent complete remission is not a more profound criterion when compared to conventional complete remission. This is why I continue to believe that the criteria proposed by the International Myeloma Working Group in 2016 was clearly needed, and I think it was a major step forward in multiple myeloma. As many will be aware and remember, there are 4 different criterion: sequencing, MRD negative, flow, imaging, and the sustained MRD negative criterion. And I will leverage on the fact that the previous presentation was in AML. Both the IMWG and the European Leukemia Net developed this criteria more or less at the same time, 2016 in myeloma, 17 in AML. But I think there is a clear singularity and advantage in myeloma, which is the fact that these criterion are intrinsically associated with methodologies that define sequencing, flow, and imaging MRD negative. And I think this was fundamental to bring some level of standardization to the field. Truth is that 5 years have elapsed since those criteria, and nowadays we have plenty of data showing how PET-CT is informative in clinical trials, but also in clinical practice. Next generation sequencing using the adaptive platform has been implemented in a very large number of clinical trials showing very favorable results in terms of patient certification, MRD negative and positive, and to some extent, these data have been reproduced in clinical practice, although clinical practice is still reduced to a low number of hospitals around the world. The other methodology is next generation flow cytometry. Perhaps its implementation in clinical trials is reduced, is lower when compared to NGS, mainly for practical reasons, but the results are also very, very relevant, using NGF to stratify the risk of progression in patients with multiple myeloma. And I must say that the development of NGF by the Euroflow Consortium has been, I would say, a landmark in terms of a single flow cytometry assay being standardized across many different laboratories worldwide. And it is important to note that the experience in those clinical laboratories using NGF is also excellent and reproduces data from clinical trials. As you may be aware, for many years there was discussion about who was the winner, which methodology was the most sensitive, either sequencing or flow. And I think that for many years, sequencing appeared as the most sensitive method. However, it is also true that at the beginning, next generation sequencing done retrospectively on samples that were stored in the respective biobanks was being compared with old low sensitive flow cytometry. And truth is that more recent comparisons between NGS and NGF show high concordance both at quantitative as well as qualitative MRD assessment, MRD burden, and patient classification into MRD negative and positive. And nowadays we see that once we have evolved from traditional multi-parameter flow cytometry into NGF, the ability of NGF to predict risk of progression and or death, that is PFS, is very similar to NGS. And in fact, I think that the most important lesson we learned over the years is that in myeloma, we need to be extra sensitive, and clearly prediction of outcome will improve if you are able to achieve a limited detection, not only of 10 to the minus 5, but even 10 to the minus 6. Throughout or in more recent years, and leveraging on new study design in clinical trials that have progressively implemented a higher number of MRD assessments. Also, treatment is now longer in multiple myeloma as compared to 10 years ago. We have recognized that MRD is much more dynamic than what we were appreciating 10 years before in older clinical trials with just 1 or 2 MRD assessments. And it's perfectly possible to see patients such as this one, in which at the time of CR there is clear persistent MRD. After finalizing all induction cycles, these were 6 courses of VRD, the patient was MRD negative, also at day 100 after stem cell transplantation. The sample is of high quality because we are seeing here mature normal plasma cells. However, only 2 months after 2 courses of consolidation before maintenance, the patient was again MRD positive 10 to the minus 5. If we would have stopped here, the patient would be erroneously classified as MRD negative. This is why, in my opinion, and beyond the method you choose to adopt, the most important criterion is the one of sustained MRD negativity. This is associated with exceptional outcomes, as shown recently at ASH by our Italian colleagues. And more importantly, once you identify patients with sustained negative MRD, it doesn't matter if you use flow or sequencing. The results continue to be very, very similar. Therefore, I think that we are living unprecedented times in myeloma, particularly regarding MRD detection. And nowadays we have multiple techniques that measure multiple features of disease biology inside and outside the bone marrow. More recently, we have mass spectrometry as a high sensitive tool to measure M protein in the serum, NGF that measures or identifies phenotypically aberrant cells both in blood and the marrow, NGS that measures clonotypic cells also in blood and the marrow, recognizing that both NGF and NGS are much more sensitive in the blood when compared to marrow, and also PET-CT that measures active cells that uptake FDG either inside or outside the bone marrow. If we have so many different techniques measuring different features of disease biology, I think it is fair to expect some level of disagreement among MRD testing methods. I think that this should not be an alarm. Rather, I believe that we should leverage on those disagreements to identify patients with exceptional outcome based on a double or triple MRD negative result. And I think this is well illustrated in these two studies. Here, the left part of the slide, you can see how patients with negative MRD by NGS and mass spec have a very favorable progression-free survival, and the same applies for patients with double negative MRD by flow and PET-CT per the experience of investigators in the University of Arkansas. There is also disagreement, not only related to methods to assess MRD, but also between conventional and MRD response criteria. For more than 10 years, we have seen how a small but consistent percentage of patients that are MRD negative show nevertheless an M protein detectable either by immunofixation or electrophoresis. Now, we have seen study after study how the outcome of these patients, identified here in light blue, is exceptional, and it is identical to all those patients that are also MRD negative and in complete remission. And therefore, a negative MRD, despite a positive immunofixation, should not be regarded as a false negative result. Traditionally, this has been explained by the long half-life of some immunoglobulins. I think this phenomenon can explain part of discordances, and more recently at ASH, our group reported data that could perhaps help us to understand other part of the discordance. And the data suggests that in patients with undetectable MRD using NGF before and after transplant, therefore reproducible, sustained, it is possible, once you isolate mature B cells and normal, phenotypically normal plasma cells, to detect the same immunoglobulin gene rearrangement as the one present in myeloma cells studied at the time of diagnosis, some of which present in surprisingly high percentages among each cell type. However, these clonotypic cells are not myeloma cells. They may be pre-existing cells, that pre-leukemic cell Dr. Dillon was talking before, or a cancer stem cell, but we also show that when we compare the mutational profile in the genomic landscape, indeed, of all these cells, starting at the CD34 progenitor, and all these cells, I insist, were collected from patients with double MRD negative, 10 to the minus 6, and therefore no possibility whatsoever of contamination during isolation. We see that these cells may share mutations with the myeloma clone, but we never find driver mutations and never copy number alterations. In fact, we believe that what we are seeing is that myeloma patients have somatic mutations in the B cell lineage, likely before the disease onset, that this mutated lymphopoiesis may increase risk of developing B cell and plasma cell oligoclonality, which precedes secondary driver mutations or copy number alterations, leading to the expansion of the myeloma clone. And we are now wondering if it is possible that these cells secrete the same immunoglobulin as myeloma cells, and therefore lead into these discordances between MRD and immunofixation, and now more recently, mass spectrometry that will be even more sensitive to detect these immunoglobulins. Now, from the prognostic point of view, and after intensive research efforts in the last decade, I think that the role of MRD as a prognostic marker is clearly well established. At the end of the day, it measures the direct effect of treatment on tumor cells. It captures patient heterogeneity in terms of tolerability and biology and response to treatment. It even recalibrates patients' risk after treatment. And I'm showing here data to illustrate this concept. We have learned recently that risk is dynamic and that patients with adverse prognosis, such as, for example, the revised ISS-3, may shift into a favorable one upon achieving deep responses to treatment, and the opposite also holds true for patients with revised ISS-1 or 2 if there is persistent MRD, let alone the dismal outcome of patients with revised ISS-3 persistent MRD that have a median PFS of 1 year, median OS of 2 years, and should be candidates to alternative treatment strategies. A dramatic tumor reduction translates into prolonged survival, and I believe that we can now say that negative MRD is the most powerful prognostic factor in myeloma. This has been extensively shown, and I think it is very well illustrated in this recent large meta-analysis published by Dr. Munshi in Blood Advances in 2020 that clearly establishes the role of MRD in all disease settings: transplant eligible, ineligible, and even relapsed refractory myeloma. In each of these settings, these are two completely different myelomas: MRD negative, MRD positive. All this evidence doesn't mean that we have learned everything. There is still a lot to be learned. And this is why I think that for the time being, the most adequate role for MRD is within clinical trials. Also, as a biomarker to better characterize patients in routine practice, but to learn how to guide patients' treatment and optimize patients' management, I think that for the time being, the place for MRD is yet in clinical trials. And I will provide a few examples. Again, last year we showed that undetectable MRD was of similar significance in terms of PFS before and after high-dose chemotherapy. Does this mean that a patient with a negative MRD after induction could be spared from immediate autologous stem cell transplantation and, for example, reserve CD34 positive cells for late relapse, let's say, reappearance of MRD? This may sound attractive, but it needs to be demonstrated in clinical trials. Another very good example is maintenance. For many years, it has been postulated that MRD could be a good biomarker to guide duration and intensity of maintenance, particularly to stop maintenance in patients with negative MRD. Well, in 2018 and 2020, there was data reported by the French and the Spanish groups, the French using NGS, the Spanish using NGF. It doesn't matter, because if you follow the criteria of the IMWG, the results are reproducible, and the results show that clearly patients that benefited the most from maintenance were those that started maintenance being MRD negative. And in fact, in both protocols, in which maintenance was stopped after 1 year in the IFM 2009 and after 2 years in the Spanish 2012 clinical trial for transplant eligible, it appears that the risk of progression slightly increases among those with negative MRD. Yet a lot to be learned. And this is why I believe we need more and more of clinical trials such as this one, the master trial. There are many ongoing. This is just one out of several examples in which patients achieving double MRD negative, 2 consecutive MRD negative results enter a period of treatment-free observation and active MRD surveillance. And I think this is very attractive, not only to enable the possibility of a treatment-free period without increasing the risk of relapse, but also to explore new opportunities. For example, if MRD reappearance justifies early treatment intervention. Certainly, MRD reappearance in patients in CR and achieving a prior MRD negative result will anticipate a clinical relapse in most patients, and this clinical relapse happens in a period of time, approximately 3 years, that makes it attractive to think about early treatment intervention. However, will it improve overall survival? No one knows until a randomized prospective clinical trial is conducted. So where do we stand in 2021? I think that what is clearly established in 2021 is that undetectable MRD is the new endpoint of treatment for patients with multiple myeloma. And I would say all patients. For example, those with high-risk disease characterized by abnormally high level of circulating tumor cells in peripheral blood. This is data that will be presented at EHA in a few weeks' time. And in these patients with high CTC levels, we show that achieving CR or even stringent CR is not enough, and only undetectable MRD is able to, at least from a statistical point of view, abrogate the poor prognosis of this high-risk feature. The other classical feature of high-risk myeloma are cytogenetic alterations, 414, 1416 translocation, and or deletion of 17p. And also at the beginning of the year, our group showed that if patients with high-risk cytogenetic abnormalities are able to achieve undetectable MRD 10 to the minus 6 using NGF sustained over time, then their outcome becomes superimposable to the outcome of patients with standard cytogenetic abnormalities that also achieved undetectable MRD. And then an endpoint for elderly, I would say even the very elderly. We showed a few years ago that even patients older than 75 that were able, smaller group, but were able to achieve negative MRD enjoy from longer time to progression as other patients with an age in between 65 and 75 years. I think that the value of MRD in the elderly non-transplant candidates is now unquestionable after the data collected in the Maya and Alcyon clinical trials that clearly shows that it's a tremendous factor to stratify risk MRD negative, MRD positive in both clinical trials. Obviously, as an endpoint in transplant eligible, and I would even say at the beginning of therapy, during treatment intensification, but also at later stages, for example, during maintenance. And both the French group in the IFM 2009 and more recently in the Tourmaline MM3 and MM4 clinical trials, and this data will be presented also at EHA in a few weeks' time, it has been shown that outcomes are very favorable to those patients that achieve MRD negativity only during maintenance therapy. In fact, it is equal to the outcome of those patients that started maintenance being MRD negative. And this usually takes place during the first year of maintenance therapy. If the patients do not convert from MRD positive to negative, then outcomes, as you can see, are significantly inferior. An endpoint in newly diagnosed and more recently in relapsed refractory myeloma with the advent of the novel agents. But I think that is a very important lesson from this data that was published recently in JCO, which is the fact that in the Pollux and Castor clinical trials, really or truly, those patients enjoying longer progression-free survival were those with sustained negative MRD result. An MRD negative result that was not sustained for a period of 12 months was not associated with long-term progression-free survival. Therefore, and in conclusion, I would say that a positive MRD test is nowadays a very reliable result. It predicts relapse in approximately 80% of patients in 3-4 year time. Also, if a positive MRD result is observed in a patient that at diagnosis already had high-risk features such as revised ISS3 or any form of extramedullary disease, then this would be an ultra-high-risk phenotype, and PFS and OS are dismal. And I think that is nowadays more and more discussion of whether or not to consider a positive MRD as a suboptimal response and a trigger of a new line of treatment. Now, for sustained negative MRD, this outcome is less reliable. One is not entirely sure if the sample was of high quality, if there was not hemodilution, or even if the sample is high quality, if that single bone marrow aspirate is representative enough of a disease that is characterized by patchy marrow infiltration and even extramedullary disease. But I think that if you consider a negative MRD with a state-of-the-art method controlling for the quality of the sample, and if it is sustained over time, then it becomes much more reliable and it enables the identification of a subset of patients in whom the risk of relapse is lower than 5% per year. In fact, it becomes so powerful that it abrogates the poor prognosis of a high-risk myeloma features. It is nowadays considered treatment endpoint for patients that are fit for intensive treatment, and we now must learn how to leverage on MRD to offer safe treatment-free intervals. I'll stop here. Thank you for listening.
Thank you so much, Dr. Paiva. That was fascinating, and so many parallels with the MRD talk from earlier on by Dr. Dillon as well. So questions, Dr. Jim O'Mell is one of our scientific advisory board in the BCNI, and he's asking how a patient in long-term sustained remission should be evaluated, what MRD testing, how often. He's also asking about imaging for a patient who has received no myeloma therapy for 10 years. So we all have some of these patients, many of them would have had VAD and a transplant in the 90s, and we probably haven't done a bone marrow for 10 years on them, let alone MRD. So perhaps you could address Dr. O'Mell's question.
Thank you. That is a very important and difficult question. Let me start with imaging. I would strongly recommend imaging alongside cellular MRD assessment, particularly if the patient at diagnosis has any sign of extramedullary disease, even a mild one, such as, for example, extraosseous plasmacytoma, because even if potentially patients with elevated LDH levels as a surrogate of possible extramedullary disease that may or not be depicted by an imaging method. In this subset of patients, or this was a subset of patients in which we have observed that there is likelihood of a false negative MRD result by flow, and I would suppose that would happen as well for sequencing. Now, in a scenario or in patients with long-term survival, and one would expect a very good response to treatment favorable disease biology, I would expect that imaging would be most often negative. And because either flow or sequencing are more sensitive than PET-CT, you need to rely on those methods to monitor MRD in the marrow. If it's negative, I would suggest performing a new assessment after 2 years.
2 years.
If it's positive, the risk will be there, and I would suggest eventually repeating the test after 1 year. And what we are now working is to develop more sensitive NGS and NGF tests to enable you the possibility to monitor MRD within that 2-year framework in peripheral blood, let's say, for example, every 6 months. I think this will be a reality in a few years' time.
In a few years' time. And we have a few more questions. So just related to that, you were addressing the frequency in patients who've been maybe not untreated for many years. But Dr. Vitaly Mikatev, my colleague here in Cork, is asking, in your opinion, what's the optimal frequency and should it be the same in all patients? So I imagine he's thinking about the earlier in their disease treatment patients who are maybe on maintenance therapy or considering stopping.
Well, I think that early on, MRD assessment should be performed and should inform on potential management. Let's say, for example, at day 100, in our hospital, it is common practice if a transplant-eligible patient is MRD positive at day 100 to offer consolidation with the same induction regimen, with the aim of trying to have the patient being MRD negative before starting maintenance, as we are aware that starting maintenance being MRD negative is clearly associated with better outcomes. So early on, I would position MRD assessment always in a patient in complete remission and position around those time points that may help you on modifying patient management. Later on, and we are thinking on in maintenance or observation in a younger patient or continuous treatment and full progression in an elderly patient, I would suggest yearly in MRD positive. Usually if it's MRD positive, the relapse will be there in relatively short time. If a patient is MRD negative, I think that 2 years is relatively safe.
Mm-hmm. And Dr. Vitale has a follow-up question, which I think you've answered really, which is about the difference between the frequency of monitoring MRD in high risk versus low risk patients. So I think it's obvious from what you've said that if you're going to be making decision points more often, you might want more often MRD.
And let me emphasize the importance on high risk patients, because for years we have seen that even though high risk myeloma patients achieve more or less the same rates of complete remission, it has never translated into the same outcome. It was always inferior. Now, for a negative MRD result to translate into identical outcomes between standard and high-risk myeloma, that MRD negative result needs to be very sensitive, 10 to the -6, and sustained over time. Because if there is any chance of a false negative result, the risk of relapse in that patient with high-risk myeloma will be much higher when compared to standard.
Thank you. Ava Segedzi is asking, could the reliability of MRD be increased by measuring multiple biomarkers? You've already alluded to LDH, but you might want to comment on that.
Well, I mean, from a methodological point of view, it would be difficult to envision better markers than the ones we currently have, because these are, I mean, the immunoglobulin gene sequence, the aberrant phenotypes by flow, these are not influenced by clonal selection or clonal evolution. These are very stable. And from a clinical point of view, I would insist that now, according to our experience, patients that at diagnosis show any sign of potentially higher probability of extramedullary disease, such as an extraosseous plasmacytoma, elevated LDH levels, also higher levels of circulating tumor cells that could lead into more dissemination and potentially extramedullary disease. In these patients, one should recognize or expect that negative MRD result may not be that reliable.
Mm-hmm. So the prior probability coming into your assessment of borderline or a positive.
Excuse me?
And can I ask you from my own? Can I ask you just from my own point of view? I was wondering, obviously the cost and complexity of this methodology is large, and when realistically do you see it being used in standard of care in countries that are yet to catch up, such as Ireland?
Well, I don't think Ireland is performing poorly than a country like Spain, for example. Might be performing even superiorly in terms of economics and the economics related to patient care. And I truly think that it should not be an argument used against assessing MRD. For example, the cost of MRD by flow in Spain is around 250 euro. And this is inferior as compared to cytogenetics. And no one is questioning the role of cytogenetics, even though in many patients, cytogenetics do not impact on patient treatment. And both the cost of cytogenetics or MRD are not comparable by all means with the cost of a single induction cycle with, for example, a three-drug regimen. So I think that if you recognize value on the biomarker, cost should not be an argument against using the biomarker.
So on that note, thank you so much for your talk, and we really appreciate your time, Dr. Paiva, today. And I'd like to thank you on behalf of the BCNI and the audience, and we'll move to the next talk, if that's okay. So, lovely. So I'd like now to introduce Dr. Mairead O'Connor, who's a research fellow with the School of Public Health here in UCC. And she formerly worked with the National Cancer Registry. She has an interest in HPV and cervical cancer, and she's coming to talk to us this morning about the application of qualitative research methods in assessing the needs of the lymphoma patient. So thank you very much, Mairead, for joining us.
Hi, Mary. Thanks. And yeah, thank you. And thanks for giving me the opportunity to talk a little bit about using qualitative research to understand the needs of lymphoma survivors. So as Mary said, I'm a research fellow, well, in the School of Public Health. My background is in cervical cancer screening and HPV infection, and I've done quite a good bit of qualitative research in that field. But now I've moved a little bit into the area of blood cancers and lymphoma cancer. So I'm just going to give a brief overview of qualitative research, what it is and how it can be used to understand the needs of cancer survivors. Then I'm just going to talk a little bit about a study we have planned, or it's in the process of being conducted, I suppose, or it's just about to start. We're just about to start patient recruitment. So first of all, what is qualitative research? Well, I suppose it's the study of the meaning of the world from the perspective of the research participant. It very much relies on words rather than hard data, and involves collecting and analysing non-numerical data to understand concepts and experiences, and can be used to gather insights into problems and generate new ideas for future research. Why do qualitative research? I suppose it's to gain an understanding or an insight into people's reasons and motivations for doing certain things. So, for example, why do parents refuse to have their children vaccinated? And also to explore the thoughts and feelings of study participants that may not be captured through quantitative methods such as a questionnaire. So, for example, how do cancer patients feel about the care they receive when undergoing treatment or in the post-treatment setting? As with any research method, there's advantages and disadvantages, or strengths and limitations to the particular research method. In terms of qualitative research, the main strengths includes that often relies on the unspoken data. So you're looking for visual cues of the research participant to provide really useful information for your study. It allows to gain perspective and views from different viewpoints of a diverse range of study participants, and it provides in-depth, detailed information. I suppose it has limitations as well, and one of the main limitations of qualitative research is the generalizability of a single qualitative research study and extending the findings to a wider population. I suppose from experience of conducting other qualitative studies, some of the other limitations include interviewer fatigue, participant fatigue, and the interviewing experience can be quite tiring, interviewer bias, and then replication issues as well for future studies. The four main types of qualitative research methods, with the most commonly used interviews and focus groups. In terms of interviews, you have in-depth or semi-in-depth interviews. These can be one-to-one or they can be dyads, which involve one or more than one participant. A lot of research studies would use face-to-face interviews, but I suppose now in the era of COVID, people have been moving to online interviews and telephone interviews as well. There's also participatory action research and observation qualitative research, which would have been used in anthropological and sociological studies before. So just as I mentioned there in the last slide, in the age of COVID, researchers have had to move their qualitative research studies online to Skype and Zoom interviews and Skype and Zoom focus groups as well. And there's also a way of conducting qualitative research through email interviews and then telephone interviews. As with face-to-face interviews, these online methods and telephone methods have advantages and disadvantages. And I suppose it's about weighing up the options of the most beneficial or appropriate method for conducting the research in order to answer your research question. So in terms of uses of qualitative research, it's quite useful for developing healthcare, behavioural change and psychological interventions. And so, for example, a self-management intervention for lymphoma survivors. It's particularly useful for researching sensitive issues that you may not be able to capture the full meaning or the person's full views in a quantitative survey. And so, for example, if you're investigating depression and sexual behaviour or sexually transmitted infections, qualitative research can really draw out people's views and thoughts more so than quantitative. It's useful for informing survey development, and it's useful for informing follow-on research studies. In terms of qualitative research and how it can contribute to the BCNI, it can complement clinical data collection and can be used to help inform patient-reported outcome work among blood cancer survivors. And it's useful for the first step in designing a patient-reported outcome study. So in terms of how qualitative research can be used to understand, I suppose, cancer survivors and lymphoma cancer survivors, just by way of background on the current situation as of 2019 in Ireland around the needs of cancer survivors. In 2019, I was involved in a scoping review, which was led by the NCCP, the National Cancer Control Programme, and the Irish Cancer Society, that looked at the unmet needs of cancer survivors in the post-treatment setting. And the aim of the scoping review was to understand the current situation in Ireland in relation to unmet needs for those living with and beyond cancer, and to perform a gap analysis on unmet needs. Is there any data lacking on specific unmet needs or for cancer types? For the scoping review, we looked at qualitative research studies and quantitative research studies conducted in Ireland in the post-treatment setting for cancer survivors. And we found that some domains were neglected, such as patient-clinician communication, family-related needs, cognitive needs, and spiritual needs. and there was no real evidence of unmet needs specific to cancer type, but we assumed that the study numbers were too small. I suppose what's pertinent to the current presentation, research on unmet needs of some cancers were lacking. Most of the studies we included in the review were on the more common cancers such as breast, colorectal and prostate cancer. I think in terms of lymphoma, I think there was only one study included in the review, and that was a while ago. So there certainly is a gap in Ireland to conduct research around the needs of cancer or lymphoma cancer survivors. And there's also very little evidence on the experiences and views of cancer patients in general around their self-management of symptoms and side effects. So I suppose just on to the current study, or the study that we're about to undertake, which is Living Beyond Lymphoma Cancer, and it's a in-depth qualitative study on the experiences of self-management preferences of patients, and it's been conducted with Professor Mary Cahill and Dr. Derval O'Shea at CUH, myself and Professor John Brown at the School of Public Health in UCC. And just for a little bit on the study background, the needs of lymphoma survivors are in the post-treatment phase are poorly understood, as I outlined earlier. Little is known about survivors' views and preferences for self-management of symptoms and treatment side effects. And by using qualitative methods, we'll be able to have a deep understanding of issues that are important to patients, which can help inform future studies. So the study aims are to explore the patient's support needs after treatment completion, gain insight into patient experiences and views on their self-management strategies. And the secondary aim is to explore the perspectives of family members or caregivers on the needs of cancer patients and their self-management strategies. In terms of methods, we are conducting one-to-one interviews among patients attending clinic appointments who have been attending clinic appointments at CUH hematology clinics. Patients with low-grade non-Hodgkin's lymphoma who are 6 months to 5 years post-treatment will be eligible for the study. And we anticipate the interviews will be conducted online via Skype or Zoom or over the telephone at the preference of the patient. We're hoping to recruit approximately 15 to 20 patients in total, which will include patients and family members and caregivers. So just a little bit about the study design. Patient lists will be reviewed by Professor Mary Cahill and Dr. Derval O'Shea, and we will purposely sample patients from these lists to write out to. And we will sample patients by age, so we're sampling by whether they were over 65 or under 65, and rural and urban area within Cork County, just to get a diverse range of views and experiences of different patients. We're hoping to send out invitation letters soon to patients, and those interested will return a reply slip to the research team, and myself will arrange a suitable interview date and time with the participants. Data collection, I suppose interviews will be aided by a topic guide, which is standard procedure in qualitative research studies. The sections of the topic guide include questions around their patient's background, their symptom burden, the effects of treatment, and with a particular emphasis on self-management. So we're going to ask them around their understanding of self-management, experience of using self-management strategies in the past, and their opinions on using self-management in the future, any barriers and facilitators to self-management, and supports needed going forward in order to do self-management. We'll also ask them a little bit about information and support seeking, and then a little bit about impact of the COVID pandemic on cancer care and help seeking. We're hoping to, we will record, transcribe and anonymize all the interviews, and the coding and analysis of interviews will take place with the aid of a qualitative data analysis software known as NVivo. And we're hoping to undertake a thematic analysis, which the main themes will be identified, which are important to the study participants. what will the study findings be used for? Well, we're hoping they'll inform practice around care and self-management in lymphoma survivors. They may help in the development of a future self-management intervention and to inform the development of patient-reported outcome survey among blood cancer survivors. So in terms of the study progress to date, we have the study protocol written and we've obtained ethical approval. We have our interviews topic guide and all other study documentation letters finalized. The patient lists are currently being reviewed and we're hoping to start contacting patients and conducting interviews in the next couple of weeks. So I'd like to thank the rest of the research team that's listed here on this slide. An apology for the short talk. We just don't have any data yet to present. So hopefully we can update you in the future. Thank you.
Thank you very much, Mairead.
Okay.
We censored your video so that we maximize the bandwidth, but I guess we could censor your slides now and bring you back on video. Thanks, David.
Okay, perfect. Good. Okay.
So I would welcome any questions from that. And just to say, I suppose qualitative research is not one that we get trained much on in medical school. And, you know, lots of people who've done science degrees don't really have much familiarity with the methods and aims. But when you discuss with the patient that you're going to do this research, they're normally very interested. And in fact, I've got verbal consent from 3 patients already, so they'll be coming back to you next week with their forms. And the attitude, I think, was, you know, oh, somebody is interested in what I have to say or what my carer has to say. You know, this is really good. So
Okay,
I think we need to remember that. And your initial slides just, I think, very much reflected that different kind of viewpoint that we need to think about from time to time as we plow through our kind of, you know, medical advances and getting the patient better medically. Are there any comments? Anybody want to ask Mairead any questions? Mairead, from your previous experience with the cancer registry, have you encountered any kind of difficulties or resistance in the patients' groups once you, or the patient's individual, once you actually get to talk to them?
Well, I suppose it depends on the participant being interviewed. I suppose there's a tendency for interviewees to go off topic, so it's kind of hard to bring them back to the main topic of that you're trying to study within the remit of the interview. And I suppose some patients have become upset, but we have procedures for handling. You know, we have a protocol normally in place for handling distress within interviews, which we have for this study as well. So
yeah. Okay, so we have a comment from Rebecca Amet, who says, nice to see this kind of research being carried out as part of the BCNI. So that's nice. Thank you, Rebecca. Okay, so if there's no further questions or comments, we'll thank you very much, Mairead, for coming on this morning and for broadening our horizons a little bit. And we will move to the next talk. So I believe we have Dawn Swan standing by somewhere. So Dr. Swan is an SPR in St. James's currently, and she's in. Yes, here we are about an long-term follow-up and analysis of the clinical trial, which of course is our flagship clinical trial in the BCNI. So welcome to the podium, Dawn. Are you there? Super. Thank you.
I'd like to thank the organisers for the invitation to speak today. My name is Dawn Swan and I'm a haematology SPR currently working in St James's Hospital. I'm going to present the updated results from the BCNI Sybel-Dara trial in newly diagnosed multiple myeloma. Sybel-Dara is a combination of the CD38 monoclonal antibody daratumumab with cyclophosphamide, bortezomib and dexamethasone. Daratumumab is an IgG monoclonal antibody targeted against CD38, which is expressed on multiple myeloma cells. It acts through several mechanisms: complement-mediated cytotoxicity, NK cell-mediated antibody-dependent cellular cytotoxicity, and macrophage-mediated antibody-dependent cellular phagocytosis. Daratumumab is given with Cybord, a regimen of the alkylator cyclophosphamide, the proteasome inhibitor bortezomib, and dexamethasone. Cybord is the most frequently used induction regimen for multiple myeloma currently used in Ireland. Enrolled patients received 4 cycles of Cybord-Dara induction, followed by stem cell mobilization and autologous stem cell transplantation. 2 further cycles of Cybord consolidation were followed by maintenance with monthly daratumumab or daratumumab and 2 weekly bortezomib in high-risk patients until progressive disease, intolerance, or a maximum of 12 months maintenance. 18 patients were enrolled on the study. The mean age was 54 years with a median of 56.5. 61% were male, and the majority had low-risk disease by traditional ISS risk stratification. 28% had high-risk cytogenetics or gene expression using the SKY92 gene expression profile. 11% had 17p deletion, and 5.6% had either translocation 414 or 1416. 11% had translocation 1114, which is an intermediate risk marker. Response data has already been presented and published for the induction, post-transplant and consolidation time periods. The rate of CR or greater rose from 11% to 44% post-induction to post-transplant, and rate of VGPR or better rose from 61% to 83% at the same time frame. I'm currently awaiting the end of treatment response assessments. I was supposed to have these for this meeting, but sadly this has been delayed by the cyber attack on the HSE. However, I do have the end of treatment MRD analysis. MRD assessments were performed in 15 patients after induction, transplant, and consolidation. 3 patients were not assessed as they had come off study. Of the 15 patients tested, 2 did not have a marker to follow, and so results are available in 13 patients. At end of treatment, 11 patients underwent MRD testing. 2 patients had progressed and 2 remained on study but missed MRD testing, which was due when the NUIG lab was shut during the initial COVID pandemic and lockdown last year. One of the tested patients had no marker to follow, so results are available in 10 patients at end of treatment. Using an intention to treat analysis, rates of MRD negativity to a sensitivity of 10 to the -5 were 17% after induction, 56% post-transplant, and 39% post-consolidation and at end of treatment. Sustained MRD negativity was achieved in 33% of the cohort, and of those who remained on study, 54% were MRD negative at the end of treatment time point. Of 10 patients who were MRD negative after transplant, 7 remained MRD negative at end of treatment. 1 did not have MRD assessment but remained on study, and 2 became MRD positive after consolidation, of which 1 progressed. Of 7 patients who were MRD negative after both transplant and consolidation, all remained on study, with 6 out of 7 still MRD negative at end of treatment. This may have been 100% if the final patient had been able to have their MRD assessment. It should be noted that the sustained MRD negative rate of 33% achieved in this study compares favourably to larger studies of DARA-based regimens, such as DARA-VRD, which reported a 12-month or longer MRD negative rate of 29%. Five patients in the study had high-risk disease by FISH or gene expression profiling. 3 came off study. 1 had deranged LFTs unrelated to the study drug. One patient died of bilateral pneumonitis during induction in a VGPR at that time. And one patient who was MRD positive post-transplant had progressive disease and died during the consolidation timeframe. 2 high-risk patients remain MRD negative to a sensitivity of 10 to the -5 at end of treatment. One of these has 17p deletion and the other has translocation 1416 and a high-risk gene expression profile. So to conclude, Cybert-Dara is a well-tolerated and highly active regimen in multiple myeloma. No patients came off the study due to adverse events related to study drug. Response assessment at end of treatment is currently awaited, but the MRD results are highly encouraging. One patient with translocation 1114 was refractory, but is subsequently doing very well with targeted therapy and venetoclax, and 2 patients who were MRD positive either post-transplant or after consolidation progressed during consolidation. MRD negativity after transplant and consolidation seems predictive of sustained MRD negativity at end of treatment. No patient in an MRD negative remission after consolidation progressed. Sustained MRD negative responses are also seen in high-risk disease, which is extremely promising as this remains an unmet cohort. Thank you for your attention, and thank you to all of the people who have made this trial possible.
Okay. Lovely. So thank you very much to Dawn there in her absence, and we can't ask Dawn questions directly, but there are quite a few people on here who participated in the Cybert-Dara study, so I'm not sure if anyone wants to make any comments. It is nice to hear that no patient who was MRD negative after consolidation progressed, and that very much chimes with what Dr. Paiva was saying as well. Anybody got any comments? Any comments to anybody? Okay.
Maybe I think we can progress, wrap up this session, and then we meet again at 2 o'clock.
Lovely. So I'd like to thank all the speakers from this morning. We have managed to come in slightly ahead of time, which is always nice. So that gives everyone a little extra time at lunch, which might be nice from a HSE point of view if anyone's dealing with the cyber attack. So our scheduled lunch break is until 2 o'clock. So I guess, Ava, we'll meet again at 2. Yeah. So we look forward to seeing everybody then again.
Thank you everybody to attending this morning, and we hope to see you in the afternoon, and that session will focus on lymphoma. So hope to see you all there back. Bye, bye-bye.
Bye-bye, bye-bye.
It'll be my talk and not Tony's talk.
You know how to share the screen and all that, yeah?
No, I don't. I share screen.
When the time comes, yeah, click share.
So,
Tony, do you want to help Elisabeth?
Sorry?
That's the share screen.
Yeah.
Yeah. And then share, yeah.
I think I'm in. I think I can manage from now.
Yeah.
Right, sure. Just down there. Yeah, whatever we usually do. Yeah,
Is this one? Is that
no, next one. That one.
Okay, I know.
That's it.
There you go.
Yeah, no, it's perfect.
I'm going to get my vaccination. I'll listen to you.
No, it's all right. They're all telling me why they can't listen to me.
It doesn't matter. The 38% should last.
Anyway, tell me when I should. Oh, I'm fine. Anyway, it's nice to see you, Philip.
Yes, lovely to see you, Elisabeth.
I've seen you for about a year and a half.
I know this damn virus.
I know.
But you still hit hard with the other type of virus, the computer virus.
We're not too bad. The way it's affected us is that they've put a cyber a firewall around St. James's that we can't get out of. So
yeah,
so everything. So if you're having a Zoom's meeting or a talk like this, you have to leave St. James's.
okay. Yeah, it's very, very inconvenient.
It's just confusing. And then I don't know about Beaumont, but James's Wi-Fi is terrible. So
so we have to change a password every day.
Really?
And reboot our computers every day.
Okay. Oh, that's very strict.
But then it works. But
so you're all right in the Empire of Beaumont, but
it's not so bad. I think because we're kind of a voluntary hospital, so we're not quite as linked in with a lot of the HSE stuff.
Yeah.
Mainly the radiology that was caused the havoc.
Yeah, that was chaos. But I think we've kind of got to the end of that. Okay. No, it's sort of I don't know if you know, Eva, but one of the things that we're quite interested in is developing a national medical laboratory information system. And I tell you, this has certainly damped my enthusiasm for it hugely. I don't know.
Yes, that would have gone down.
Would have been a disaster, wouldn't it?
Yeah, but we're lucky we have a 40-year-old computer system. It's too old to be hacked. So,
I like your positivity, Philip.
Yeah, you've got to look at the bright side.
Yeah. But now we have these old-fashioned blue backgrounds, Eva. With,
like the DOS background.
Absolutely, it's exactly like the DOS. But nobody can. Yeah, maybe we'll all be back to that.
Oh, it's a good reminder, to appreciate what we have usually.
Yeah. No, it's definitely it's just been a disaster. They say it's going to take ages to resolve.
I hope it's worthwhile, and then the new system, as when it gradually opens, it will be more robust.
We're going to have to put a lot more money into cyber security. That's for sure.
That's the problem.
And it's one of these areas that it's not like a shiny new machine or a new ward. It just doesn't.
This is typical of the health. This is typical of the health service. You only do anything after the crisis has happened.
Yeah.
And there's a disaster.
Yeah.
And then they have to do something about it.
Yeah.
But anyway, so when do we kick off there, Eva? Are we nearly?
I think maybe, so we have 2 minutes till 2.
Okay.
And then we could give a few more minutes to people, just to come back on.
Yeah, that's fine. How was your lunch then?
The lunch?
How was your lunch?
I was looking at hematopoietic stem cells. We are testing out a colony forming assay. So
you're a workaholic.
Tiny colonies after 4 days of culture. It was great.
Looked good, did it?
Yes, there in some cases I saw 3 cells, so we are working out the maths. Okay.
Excellent.
From 1 to 3. I think that was
fantastic. Brilliant.
So yeah, it's we miss, the thing that I have to say you really miss is the kind of discussions that happen by chance, which are often the most interesting discussions.
Yeah.
And they just aren't happening at the moment.
No.
Hi, Elisabeth.
Oh, hi, Mary. How are you?
I think next year we do a proper symposium where we have coffee together and we walk around.
Yes.
More than coffee.
Yes, you're right.
And no cells to distract.
We definitely should do that, Eva, because it's a good formula, and people are enjoying it, and because there's the was it 96 people have signed up or something? So if we got even half that number to come to somewhere, that would be nice.
Yeah.
No, I think the morning session was very good. There were lots of people here.
Yeah.
I think it was about 80 people, Elisabeth, on the call.
Well done. Yeah. Might tail off a bit now, Friday afternoon, rainy Friday afternoon.
Never mind that. I think it was still, I think, as good as we could make it under these circumstances.
Oh yeah, no, absolutely.
And we've saved the best wine to last.
Philip, you're very kind. You always were.
Yeah. Well, I'm looking forward to this. Yourself and the other speaker look very good.
Yeah, no, absolutely. And I haven't met him, so I'm looking forward to. I'm certainly looking forward.
He's actually online. So basically,
is that me?
Yes, that's
You're talking about me.
Yes, you're talking nice things.
I'm listening. I'm listening. Yeah, I'm listening. I'm listening to
Professor Murray. That's Professor Paul Murray.
That's me. Yeah, yeah.
Who's
we can't
in the
put a
UK
face
at the
on you,
moment.
Paul. We'd like a face to go with
Shall I
the...
put the, let me put the camera on. There we go. That's yeah.
Hello, how are you?
Lovely.
Hi, good.
Yeah. No, I'm really looking forward to your talk because
well, it's pre-recorded actually. So
yeah, I heard that. Yeah.
But I was meant to be on jury service this week, but I sat there for 3 days and I didn't get picked for any panels at all. So I just felt like nobody wanted me really. So here I am, in person, as it were.
I think I would consider it a lucky escape, really.
Yes, yeah, absolutely.
Yeah. I suspect jury service isn't great fun.
No.
No.
One of the few advantages of being a medical doctor is you're absolved from it.
Is that right? Yes.
Yeah, we're never asked.
Yeah.
So how long have you been working in Galway, Paul?
I'm in Limerick.
Oh, you're in Limerick. Sorry. Yes.
Well, so yeah. I've been there about around about 2 years, but I've still got my lab in Birmingham in the UK.
Okay.
But I was saying to Philip there, we were planning to move over to Limerick last year. Obviously now that was put on hold. So yeah. So my wife has a job there that she hasn't started yet.
in UL or in Birmingham?
In UL.
Okay.
Yeah.
Oh, yeah. Right. Yeah. Yeah. All these funny, all these kind of unpredictable effects of COVID.
Absolutely. Yeah. Well, I had a postdoc there who started with me in Limerick. She was offered a post in Harvard actually, and which she decided to take. And the poor girl, she accepted the post, but then she couldn't go there, and then they terminated her contract, so she was without a job. So all kinds of, as you say, knock-on effects that you can't predict.
So the boss lady has asked me to start the session.
Okay.
So
right.
My name's Philip Murphy, and I am chairing this final session. We've had a brilliant day so far, and I'm looking forward to this one as well, which is concentrating on lymphoma. If I don't get a chance, I'd just like to thank again Eva and Sharon and Mary for all the work in putting this together. So our first speaker is very well known to us, Professor Elisabeth Vandenberghe, who's a consultant hematologist at St. James's Hospital and clinical professor in Trinity College, Dublin. And she's the lymphoma clinical lead, the hematology clinic director of the Cancer Molecular Diagnostic Laboratory, the clinical director of the Cancer Clinical Trials Unit, and associate director of the Cancer Research Facility at St. James's Hospital. And she's going to tell us all about mantle cell lymphoma. Thank you, Elisabeth.
Thank you very much, Philip, and again, thank you for the organisers of this for inviting me to talk about, yet again, lymphomas. It's a tribute that despite all the viruses lurking around, that you've actually managed to do have a symposium on a rainy Friday afternoon. So generally speaking, I think when you put the first slide up, it shows the name of the talk and then the speaker. And I felt that this really didn't reflect what I was going to talk about at all, because I'm really only one of quite a large team involved in these two projects I'm going to discuss. So from the clinical perspective, of course, I have to thank the patients who were involved, the nurses and doctors I work with, especially Larry Bacon, and of course, the national referrers who really made this project, as you'll see, possible. Richie has been really a wonderful hematopathologist to work with, as has Fiona Quinn, who's the senior lymphoma clinical scientist in CMD. So really the first part of the talk is very much based on their work, not really mine. And then James Nolan is our PhD candidate working with Adrian Bracken, who is a professor of medical genetics in Trinity. And really the second part of the talk, I really had no hand or part in, except to be amazed at how James has taken like a duck to water to being a proper research scientist, and how skillfully Adrian has taken him through a translational project. So this is very much based on what is translational medicine. It's that space between clinical medicine and basic research, and it should be a very inclusive space. So I suppose I get very upset when I hear, oh, it's bench to bedside, because I would say a lot of translational medicine starts actually at the bed. And a good clinical observation can lead to very, very strong and interesting research. So really, I don't think it's a place where one should predominate over another, and we really need to work together. And to that end, we need lots of coffee and lots of wine to make those conversations happen. And I'm just going to use two projects that I'm involved in to show this. And of course, the first is mantle cell lymphoma, which is, I suppose, since my PhD, I've been very involved in and interested in. And mantle cell lymphoma is a rare lymphoma. It's between 5 to 8% of all lymphomas diagnosed. And it's really highlighted in at least 90% of patients by translocation 1114, resulting in the immunoglobulin gene promoter upregulating BCL1, which is now known as cyclin D1, which is a cell surface or cell cycle protein. And really, the translocation 1114 happens in the bone marrow, but I suppose doesn't cause a block in how the cell matures until it gets to the mantle zone of the germinal center of the follicle, the lymphoid follicle. And then there's a sort of a little anomaly, and I'm just going to briefly talk about this anomaly because I think it's clinically very relevant. So most mantle cells are pre-germinal center. They don't undergo that germinal center education, which say follicular lymphoma or post-follicular lymphoid malignancies have. And what happens in this germinal center is isotype switching and hypermutation of the immunoglobulin gene. So when a mantle cell goes through this germinal center education, it becomes much, much more indolent. About 5% of all mantle cells do this. And then this is what I hate, sorry about this. I need to go back, thanks. So these lymphoma patients have a very indolent course and often never need treatment. But if you do a MIPI score on them, which is well known again to clinicians, they actually have a high MIPI score because they're often old, they often have very advanced disease. So just a little caveat from a clinical perspective of something that happens biologically that's a bit of an anomaly. But 95% of patients don't go through this germinal center education, and they end up as being classical mantle cell lymphoma. So that's about 85% of cases in general. And they have a predilection for the mantle zone and then take over the entire germinal center, I suppose, morphology. And then about 10% of these patients either de novo or transform into a blastoid or pleomorphic mantle cell lymphoma. And these are associated with 3 genes, mainly TP53 mutation, and then less often Notch and Notch1 mutations and KMT2D. So the clinical picture, just again, it's a very interesting lymphoma from another perspective, and that is the most marked male predominance of any lymphoid malignancy. And it's something that hasn't been studied, and I don't understand it at all. So if anyone has any bright ideas as to why this particular lymphoma happens 5 times to 6 times more often in men, I'd really be very interested in the theories. And as I've said, a predilection for the mantle zone of the Waldear's ring, gut, and spleen. The survival, when we first identified it in the late 1990s, was only 3 years with conventional treatment. And actually, if you look at HMRN data, you'll see that it hasn't improved for the vast majority of patients a lot since then. But the real problem is in relapsed patients, where the median progression-free survival of those patients is only about a year. So I'm just going to walk you through the treatment. And the reason I'm walking you through treatment for you scientists, if any of you are listening, is that the treatment actually tells you something about the biology. And that is, when we first started treating these patients, very few of them survived. But as soon as we introduced Ara-C and rituximab into their treatment, you got overall survivals of about 50%. Now, rituximab is interesting because, of course, it doesn't depend on any particular mutation profile. It just depends on CD20 expression. And Ara-C is even more fascinating because after all these years of this ancient drug, we are not completely sure of how it works. We know that it's a bit affected by TP53 mutation, but it probably is effective in TP53 mutated diseases. But the real improvement happened when we started using autologous transplantation in first remission. And again, I think we've hit a little bit of luck here. We certainly didn't know it in 2002 when I actually published the EBMT outcome from primary autologous transplants in mantle cell lymphoma. And that is that BEAM depends on not just an alkylating agent, in other words, an melphalan, but it actually uses 3 drugs that are possibly TP53 independent mechanisms of action. So when we started looking at first-line consolidation transplant in these patients, if you go back, sorry, there's some inherent timing in this presentation that I haven't gotten rid of. We see that with the Nordic protocol, we're getting overall survivals of about 50% at going out now to 12, 14 years. Now, it looks as if there's a plateau, but in fact, there isn't a plateau. What's actually happening is we're now using good second-line treatment, which we didn't have in the earlier days. So patients are continuing to relapse, even with an autologous transplant in first remission, but it certainly has improved the survival of these patients dramatically. And then if you go back to current HMRN data, we see that overall patients over 70 are still doing really badly with mantle cell lymphoma. And hopefully the use of the newer agents will change this. And again, both new therapies that I'm going to talk about are have TP53 independent modes of action. So targeted treatments with ibrutinib have been incredibly successful in mantle cell lymphoma. And I always think about targeted treatments. Why are they targeted? Well, they're targeted because each, I suppose, cell or target cell has become addicted to a particular group of intracellular pathways. And it's no surprise that B lymphocytes depend on their B cell receptor and the downstream intracellular pathways for survival. And well ensconced in there is the Bruton tyrosine kinase pathway, which is inhibited very effectively by ibrutinib and its lookalikes. And what we see from that very early study, which I think a number of us were involved in Ireland, is that patients who get ibrutinib in second-line treatment do very well. So they have about a 50% survival at 2 years. Now, clever people will say, well, it's exactly the same as the standard of care. Sorry, if I could go back. Standard of care, and that's because there was a huge number of patients who were able to cross over. So basically what you're looking at is a 100% improvement in overall survival in relapsed refractory patients when they get access to a simple targeted treatment, which is taken orally instead of all my horrible and Dr. Murphy's horrible chemoimmunotherapy approaches. And I think the most hopeful thing that's happened to us is a publication from the New England Journal late last year, which showed in a really high-risk group of patients with mantle cell lymphoma that these patients seem to have durable remissions. The follow-up is very short, but if you look at the outcome of CAR T therapy in diffuse large B-cell lymphoma and hope that you can, I suppose, use the same approach in mantle cell lymphoma, it seems that if you can get to a year in remission from CAR T therapy, you're probably fairly safe. Now, I think we'll need to follow these patients up for much longer. Now, I would like to point out that these patients, therefore, are particularly high risk. So if you look at them, all of them have failed chemoimmunotherapy, all of them have failed Bruton tyrosine kinase inhibitor treatment. 30% of them had either blastoid or pleomorphic morphology, and quite a high number of them, of those tested, had TP53 mutations. So, and yet these patients are coming out with a 50% overall and progression-free survival at about a year. So I think this is definitely the way we're looking in, we're going in mantle cell lymphoma treatment. And so I've sort of been bringing up the mood music, if you like, of TP53. And really it's been, I suppose, filtering around in mantle cell lymphoma for a long time. And I think as in CLL, we were both hampered by looking into exactly different cohorts of patients and how they behaved by the lack of availability of a good diagnostic tool to look for mutations. It's got a pivotal role in cancer, we all know that. It's the final common pathway of most resistant cancers, and the mutation confers resistance to alkylating agents and purine analogues, radiotherapy, and a whole host of other treatments we might use, but not targeted treatments, not immunotherapy, and not CAR T therapy. Sorry, I don't know what is happening to my I think probably investigating TP53 mutation status in lymphomas depended on us having TP53 independent treatments that were successful and available. And the second thing is having an available test to look for these mutations. So in June 2020, I decided to initiate a study, and I do want to tell you that it was in June 2020 and not later, because a number of subsequent publications have come out showing that I was very much in the mood, but maybe a little bit late in the doing of the project. So the Nordic cohort from St. James's Hospital is a national cohort of patients. We've been doing the same treatment and doing the same analysis on these patients from 2007 to 2019. And because they were a standard group of patients, I felt if I looked back and looked at the incidence of TP53 mutation, I could work out whether it would impact outcome. I could see what the incidence of the TP53 mutation was in this group of patients, whether we could correlate with P53 IHC, should we change our diagnostic pathway, and should we use TP53 independent treatments? And I've got the answer to some of them, but not all of these things, but I'm going to show you the results. So there were a very characteristic group of patients, median age of 59, mostly men, mostly high-risk patients, 50% having a high Ki67, nearly all extensive disease, and 10% had non-classical subtype morphology, with a variation in MIPI indexes, mainly intermediate to high, and all standard CHOP RSC induction with a beam autologous transplant. I'm going to give you two examples of how variable mantle cell lymphoma can be, even at this sort of very standard cohort. So the first patient is a 64-year-old patient from Canty who presented with a change in bowel habit, had a colonoscopy which showed lymphomatoid polyposis, was staged and was found to have widespread adenopathy. The morphology was classical mantle cell lymphoma, and the P53 and TP53 were both negative. He was treated with the Nordic protocol, and I think in 2020, when we last got his follow-up, he had survived for more than 90 months in remission. And then if you can contrast that with the next patient, who is a 62-year-old lady who presented very acutely to my colleagues in Tala. Again, she was found to have widespread adenopathy, but on this occasion, she also had a very big spleen. She was treated with the Nordic protocol, relapsed really within a year, and died very soon after. And I'm just going to show you briefly her morphology. And again, I'm really apologetic about this. So her morphology shows a typical morphology of mantle cell lymphoma with slightly nodular appearance, but a diffuse infiltrate, and then at higher power, a very uniform but slightly pleomorphic group of cells which are intermediate in size. And if you look at the p53 immunohistochemistry, it shows that it is positive. So she had a p53 mutation, and that was confirmed by NGS TP53 analysis carried out by Fiona Quinn, showing a mutation which was a missense non-functional mutation, which was with a deleterious effect. So if you look at, so the next thing we wanted to do is just make sure that our group of patients was similar to the outcome of other published series. And if you look at our overall survival at a median of 8 years, we're very much in the correct ballpark. Unfortunately, our progression-free survival is slightly less good at 6.5 years compared to the Nordic one of 8 years. And I don't really understand the reason for that. Very disappointing, but there you go, that's our data. And then James put together this comparative study of the DNA and tissue available. So we had comparable data in 30 patients. Of those, 4 patients had pathogenic TP53 mutations. 2 patients had non-pathogenic TP53 mutations, and that correlated 100% with the IHC, which was 100% mutation, 100% P53 accumulation, which is really, I suppose, felt to be comparable to missense mutations. We had 2 further pathogenic mutations in patients where we only had DNA available, and 1 further P53 accumulation where we had no DNA available. So the concordance when we could test it was 100%, but we had these 2 rather odd cases of patients who, if we had just looked at IHC, would have suggested they were high-risk patients, but when we looked at TP53 mutation status, things weren't quite so clear-cut. So for anyone who's interested in where these mutations were situated, Fiona provided me with this picture, and the variant allele frequency varied from about 5% to 80%. So I'm just going to talk briefly about these TP53 mutations of unclear clinical significance. So 1 was male, 1 was female. They both had stage 4 disease. They both had classical disease. And the P53 protein had accumulated on the IHC, but the mutation, which was a missense mutation on NGS, was felt to be non-pathogenic. And the outcome of both patients is actually reasonably good. Now, clearly 5 years is possibly not enough. And we're really going to investigate these cases first by carrying out a full lymphoid panel with NGS, and also trying to team up with the Nordic group of pathologists to see if they can identify any other cases. And I suppose what this means from saying that IHC and NGS are interchangeable, I'd have to say I don't think they are interchangeable, and I don't think you can say that NGS shouldn't be used if our treatment pathways change. And then if you look at the outcome based on TP53 mutation, there is a difference in overall survival, and less of a difference in progression-free survival, and I think the overall, again, I'm not quite sure about why this difference has occurred. I think we need to do a little bit more work on it, but it possibly reflects a slightly small patient cohort. Now, I've just talked about the Nordic protocol, which is basically for patients aged under 70 at first presentation. And of course, as we all know, mantle cell lymphoma can affect people that are far older. So I thought it would be useful to talk about the outcome of patients who are of any age who have relapsed or refractory disease. And what this very nice review, which I suggest everybody reads, suggested we do is that we divide these relapsed refractory patients into two groups. Those that are high risk, and these included patients who progress within 24 months, have blastoid or pleomorphic morphology, or are TP53 mutated, versus all the other patients. Now, in one way, it doesn't make any difference because they suggest that second-line treatment is always going to be with a BTK inhibitor. But then the patients who don't go into a complete remission with a BTK inhibitor and who are high risk should be considered at that stage for CAR-T therapy. So in fact, what we've done now is we've taken out those myriads of small studies and said, we go for broke here. If you fail BTK inhibitors and you're fit enough to have CAR-T, you should have CAR-T treatment. Now, given that it's not licensed, or certainly in Ireland, for use yet, this is a bit of a jump, but I think this is the jump that we're probably going to have to make. So how has this study changed the way we're doing things in James's? Well, I'm very happy that I consented patients and I banked DNA in most of my Nordic cohort, because in the few patients where I actually changed the MRD approach to banking DNA, and again, Mary, thank you very much for your help with this, it was actually quite difficult to go back and get DNA to carry out this study. The pathology review that Richie now carries out on all Nordic patients is FISH for 1114, SOX11, P53 and Ki67. However, the role of NGS TP53 is not completely clear. And I suppose what I would say is that patients who are IHC positive, I think we should now proceed with an NGS panel, because I think what it does, it doesn't dictate a change in management plan, but it does make you much, much more sensitive about making sure that a patient is in optimal remission before plodding on with your beam autologous transplant, especially in somebody where it might be particularly toxic in slightly older patients. However, beam is magical because I think 3 of the drugs are probably TP53 independent. I would also think that maybe all patients who have relapsed refractory disease, given that we might in the next 18 months to 2 years have a completely different management pathway for them, should be considered for NGS. And I think, you know, how can we change, knowing that patients don't do as well if they're TP53 mutated with the Nordic protocol? I think at the moment we can't do an awful lot about it except wait for this triangle study, which Ireland sadly wasn't included in, before we know the role of introducing TP53 independent treatment in induction treatment, which the study will answer. And you know, COVID has thrown up some interesting, I suppose, interesting treatment adjustments. So I've had 4 patients who normally would have just plodded through the Nordic protocol, but because of delays, because they either got COVID or because their treatment was affected by COVID, they've actually had to have their treatment interrupted. And these patients are actually being bridged to transplant with ibrutinib. And I think those 4 patients might be very informative because some of them happen to be TP53 mutated. But that'll be anecdotal information, but still always interesting, I think. So I'm going to change track completely here. And I'm going to say, I am a hematologist. I'm very interested in clinical medicine, and I'm very interested in diagnostics, but I am not a laboratory scientist. And this part of the project has nothing to do with me. And I'm really very grateful to both James Nolan and Adrian Bracken for giving me these slides and allowing me to talk about what is very much their work. And we're going to introduce a translational element into it. And the reason I'm talking about it is because we feel very much that we've become part of the genetics department in Trinity, and a very welcome collaboration it has been. So just to talk to you a little bit about follicular lymphoma, it's the commonest low-grade lymphoma we see in Ireland, about 200 new cases in Ireland annually. I think that's an underestimate because I see an awful lot of them, and I suspect Philip sees an equal number of them over in Beaumont. So they're the paradigm lymphoma of germinal central cell origin, so a step on from the mantle cell origin. and they're associated with the 1418 translocation with upregulation of BCL2, which is an anti-apoptotic gene. So if you think of cells getting arrested when their gene, I suppose, translocated gene becomes active, these follicular lymphomas, where the translocation happens in the bone marrow, they come to maturation arrest in the germinal center. And they generally have a very, very indolent disease pathway. So with current management, the Swedes have done a very lovely epidemiology study showing that patients actually have a normal life expectancy. I suspect that's only in Sweden, because a number of patients certainly run into problems. And these patients can be picked out clinically if they progress within 24 months by a POD24. But in contrast to CLL and mantle cell lymphoma, we don't have actually very good ideas. We can't, when a patient comes into us with a follicular lymphoma, we can do flippy scores and we can say, well, my intuition is that you're not going to do very well, but actually we don't know. So one of the big problems, I think, in follicular lymphoma clinical management is you can't say to a patient, you've got bad disease, until you've treated them and until they've shown that they have bad disease. So why do we have this clinical variability in follicular lymphoma? And I think it's due to three things. The secondary genetic abnormalities, secondary to 1418 translocations. And if you were like me many years ago in a cytogenetics lab trying to do cytogenetics on low-grade lymphomas, you'll find that a lot of follicular lymphomas had very complex karyotypes with very, very specific and recurrent secondary abnormalities. And that's really been proven as well in now looking at mutation analysis in follicular lymphomas. They tend to have a lot of different secondary abnormalities picked out by NGS. The second thing is microenvironment is really important in follicular lymphoma. And then the third thing, which is of course where Adrian Bracken comes in, is epigenetic modification. So if you have a POD24, that's, in other words, if you progress within 24 months, your outcome is very, very poor. So just to touch briefly on the microenvironment. So the non-malignant part of follicular lymphomas takes up 50% of the cell mass. And microenvironments mean that tumors can grow and that they can also evade the normal immune responses which stop us getting cancers every day. And they can also, in fact, allow tumors to evade chemotherapy and other anti-cancer treatments. And what Stout did in 2004, and this work hasn't really shown much, I suppose it hasn't really translated into clinical utility, but I'm always hoping it might do, is if he took out this tumor microenvironment and got rid of the tumor cells, he found that, and compared the gene expression profiles to overall survival, he came up with two very relevant immune responses. So immune response 1, where there were a lot of T lymphocyte and macrophage expressions, and these patients did well with an outcome of over 10 years. And for those who had the immune response type 2, with a lot of dendritic cell expression, they had a very poor outcome. And of course, one of the interesting things is the ASCEND study, which looks at R squared, in other words, rituximab and lenalidomide, which treats the microenvironment and the lymphoid cell together, is quite active in relapsed disease. Now, it's not as active as the drug companies peddling lenalidomide would suggest. I mean, the median progression-free survival is less than 2 years. I mean, it's not wonderful, but it does show, I suppose, proof of concept that the microenvironment is very important in follicular lymphomas. But I'm going to try and talk about the importance of epigenetics in follicular lymphoma. So this lovely study, or a review by Vogelstein in 2013, showed that chromatin modification was very important in all cancers. That's not surprising, I suppose, when you think that chromatin modification decides which genes are going to be expressed and which aren't. And EZH2 came up as one of these very important genes. And at the same time, Randy Gascoigne and Ryan Moran in Vancouver came up with the importance of EZH2 mutations in germinal central cell lymphomas, both of diffuse large B cell and follicular lymphoma subtypes. So up to 25% of cases will have mutations of EZH2. And if you look again, that work was then progressed by Jude Fitzgibbon from Barts, who has this wonderful, I suppose, biobank going back 40, 50 years of sequentially biopsied follicular lymphoma patients. And I suppose what I forgot to say at the beginning, because I keep on forgetting I might be talking to a mixed audience, is that follicular lymphomas have this, we can't cure them with chemoimmunotherapy. And over the lifespan of a patient, they can have up to 3 reactivations of disease. And if you're very organized as they were in Barts, you biopsied and biobanked each patient as they progressed over time. So they have this wonderful archive going back over many patients. And they showed that EZH2 mutations occur very early in a patient progression, so that they're a very important part, like with BCL2 upregulation of follicular lymphomas. Also of interest in the last couple of years, there's been a huge interest in trying to subdivide diffuse large B-cell lymphomas based on mutation, their mutation profile. And interestingly enough, EZH2 mutations come up as a good prognosis marker in diffuse large B-cell lymphoma. Again, saying, you know, it occurs early, but maybe it's not a bad player overall. So of course, as soon as you get an abnormality, a mutation, which happens early in a lymphoma, and the drug companies become very interested. And indeed, an inhibitor called temazestat was developed fairly quickly. And the study was, I think, first talked about in EHA last year and was published in Lancet Oncology. And what this shows is that temazestat is active in follicular lymphoma, which has relapsed after chemoimmunotherapy and is in fact in second relapse. Now, the problem about these patients is rather like with the R² story. It is active, but the duration of remission isn't stellar. So this was, again, a proof of concept that if you inhibit EZH2 in patients with relapsed disease, and they have had to have at least 2 relapses, patients do show some activity. But the truth is, we probably need less than 2 years duration of response to have a really meaningful outcome for this approach. Nevertheless, in June 2020, the FDA granted approval for this drug and in patients with EZH2 mutations. So I suppose this is a slide that Adrian gave me. It's a slightly simplistic slide, I suppose, but it does show, again, an idea that if you're going to use targeted therapies, you're going to have to treat your targets at multiple levels. And I think we know that now from using things like ibrutinib, that you can't use ibrutinib on its own. You've got to combine it with other treatments. And I think that's what's really going to happen with temazestat. And so that's really where the direction of travel of Adrian and James's work is. So they're looking at other elements of the polycomb repressor complex to see and try to identify other genetic dependencies on this PRC2 complex, and then seeing if these other genetic dependencies can be druggable. So one of the other components of the PRC2 complex is EAD, which actually does have an inhibitor, and that inhibitor has been available for a number of years. I think the fact that we haven't heard about it in lymphoma treatment circles would suggest that there are problems with it, but I really don't know an awful lot about it. And
Elisabeth,
yeah, I'm over time, am I?
A little bit over time. If,
okay. Well,
another 5 minutes maybe or something.
No,
okay?
no. I actually, when I went through my talk on my own, I was well under time, so I'll move on.
Okay, thanks.
So basically, the PRC complex has, or PRC2 complex. Sorry, the one genetic dependency has been identified by James and Adrian, and they're working on it at the moment. And they're using, at the moment, their work is dependent on cell lines. And anyone who's worked in lymphoid cell lines knows that they're notoriously difficult to work with and not really reflective of what we see in the clinic. So our current next challenge is to develop a microenvironment that is relevant for patient samples and which they can work on in the epigenetics laboratory. So in summary, I've talked about two projects with different starting points. Mantle cell came very much from the clinic, and thank you very much clinicians who referred patients in. And the second one is a pure epigeneticist who is becoming fascinated by follicular lymphoma. But they have common requirements, and I think this is why I thought it might be interesting for BCNI to talk about the two projects. So both of them need, I suppose, very specific treatment pathways, and I suppose that requires from clinicians a certain discipline. And I think we've actually been very good about that in lymphoma treatment in Ireland, and I think it's been one of the successes of the LFI. What we haven't been so good about is, because we haven't had the backup, is data capture. I think targeted consented biobanking has been, again, key to both these projects, and will become even more key for the follicular lymphoma project. I'm very lucky to work with excellent diagnostic colleagues, and I think multidisciplinary interaction has been, again, key. So thank you very much, and apologies for going over time.
Thank you so much, Elisabeth. That was fascinating. I'm sorry to have cut you short a little bit.
No.
I don't know if anybody has any questions on chat, or anybody want to. I could ask a question. Obviously, venetoclax is flavor of the month in certain conditions like CLL. Does it have a role in either of those two types of lymphoma you mentioned? Are you talking about? Interestingly,
in follicular lymphoma, it actually doesn't, which is, of course, completely unintuitive, because you would expect it to be very active because of its role in BCL2 resistance, or BCL2 blocking. And the thought is that it might be useful if you can work out blocking at multiple levels. So in other words, you might be able to get at a very early component of follicular lymphoma, but you need to have sequential blocking. And basically, I think AMVI aren't terribly interested, because follicular lymphoma is felt to have too many other treatment options, and AML is their big interest. So I think follicular lymphoma is probably a no. Mantle cell lymphoma is actually a sort of a semi-yes. And again, I think, in fact, if you look at the data that's published, it's not very good. And again, I think AbbVie are probably not very interested in mantle cell lymphoma as being a small beer compared to AML. But I have certainly used it with as a bridge to allogeneic bone marrow transplant. I've also used it post-allogeneic bone marrow transplant in minimal residual disease status, pre-DLI and pre-graft versus lymphoma effect. So I do think it's active, and I just think, I just think it's suffering from the fact that it's a small disease with a small patient, thing. Yeah.
Okay. So we've got a couple of questions. Mary Cahill says, ibrutinib in combination, as you mentioned, is better than use alone. Would you like to expand on that in mantle cell?
Well, I mean, I would always try to use ibrutinib. I have to be careful of myself now, because I'm not quite sure what the NCCP say on the matter, but I would always combine it with an antibody. You're absolutely right. In CLL, I would always try to combine it with obinutuzumab, because I think obinutuzumab is engineered for CLL. In mantle cell lymphoma, you've got very high CD20 expression, so I'm very happy with rituximab.
Great. Finally, Izat has a question. He says, thanks, excellent talk. Do you consider a young mantle cell lymphoma with T53 mutation for allogeneic stem cell consolidation?
Izat, you're making me say things in public that I might prefer to say over a cup of coffee. I think what the data so far shows us is that they're in a high risk category. And I think what I want to do now is look back on my mantle cell lymphomas that have not been eligible for the Nordic protocol because they haven't even got there. So those patients, I'm sure, are all going to be TP53 or notch mutated. So I think the problem is if you have a TP53 mutated patient by NGS with a deleterious missense mutation, I think, but they go into complete remission, it's very difficult to say to them, I'm flying against everybody else's practice and I'm going to offer you an allogeneic bone marrow transplant, but I would have that conversation with them. So, and I think the other thing is I'd really make very, very sure that that patient was in optimal remission. And if I had any concerns, I might actually do a bridge to see what their biology does. So that's putting my head on a very unevidence-based approach to life.
Okay, thank you very much. So thanks very much, Liz. That was wonderful. And
well, thank you very much. Very kind words.
You're very kind. Thank you, Elisabeth. Before I move on to our next speaker, I just want to refer back to the talk by Dr. Bruno Paiva, who looked at flow cytometry for minimal residual disease in myeloma. Just to remind everybody about that, my colleague Siobhan Vlavi is doing a big project looking at minimal residual disease using molecular means for myeloma. And we'd be very enthusiastic if other people could participate in this. And if you're interested, please get in touch with her. So let's move on to the next speaker, and that is Professor Paul Murray. His talk is recorded, but he is here in person to answer questions afterwards. He's Professor of Molecular Pathology in the Health Research Institute, University of Limerick. His lab is focused on leveraging novel insights into the pathogenesis of viral-associated cancers for the development of new therapies. His works contribute to several discoveries in the field, including the first description of C-flip-mediated anti-apoptotic phenotype in Hodgkin's lymphoma, discovery of aberrant mTOR kinase activation in Hodgkin's lymphoma, and which has led to clinical trials. So I'm going to pass you over to the recording by Professor Murray. Thank you.
Good afternoon. The topic of my talk is the microenvironment of B-cell lymphoma. Specifically, I'm going to focus on a discussion of the microenvironment of diffuse large B-cell lymphoma and hopefully provide you with some additional new insights into the nature of this microenvironment and how it might influence the genomic features of the tumor cell. So of course, we know that there are a multitude of different subtypes of non-Hodgkin lymphoma, the most common of which, of course, is diffuse large B-cell lymphoma, which itself is a fairly heterogeneous malignancy comprised of at least 3 major subgroups, the most common of which are the so-called activated B-cell type and the germinal center B-cell type. We now know that these groups, having initially been defined as on the basis of their gene expression profile, we now know that they are largely distinct in terms of their molecular characteristics and to some degree in the nature of the tumor microenvironment. Survival in diffuse large B-cell lymphoma remains a significant challenge. And despite the introduction of rituximab to combination chemotherapy, survival still plateaus at around the 50 to 60% mark, indicating that this remains a significant amount of work to do in terms of improving outlook for patients destined to die of their disease, but also in terms of reducing the intensity of treatment, which of course is associated with severe both short-term and longer-term toxicities. We know that the activated B-cell type has a poorer outcome compared to the germinal center B-cell type, shown here in terms of progression-free survival on the left-hand graph and overall survival on the right-hand graph. but there's a growing amount of evidence that the tumour microenvironment plays an important part in terms of predicting outcome, but also defining some of the molecular and biological features of the malignancy. And if we think about B-cell lymphomas, there's a spectrum of disorders representing different ways in which the microenvironment might be depicted histologically, but also from a biological standpoint. Then on the left-hand side, we can see that Hodgkin lymphoma is really a disease characterized by rather extensive inflammatory infiltrates, which one can think of really as being a disorder of recruitment and retention of lymphoid cells. Follicular lymphoma, where there's re-education, and Burkitt lymphoma, which perhaps rather naively, I guess, to a certain extent these days, might be regarded as an effacement of an existing tissue microenvironment. Diffuse large B-cell lymphoma probably sits somewhere between all three of these in terms of the nature of the tumour microenvironment. I suppose the first evidence that the microenvironment might be important was originally defined on the basis of studies done by Lenz et al., where they defined two stromal signatures, so-called stromal signatures, which were characteristic of different components of the tumour microenvironment. And these stromal signatures predicted outcome independently of subtype. Something I'm going to touch on actually as we move through the talk is the importance of the Epstein-Barr virus in a subset of patients with diffuse large B-cell lymphoma, where the virus can be detected, as you can see in the bottom two images, using EBNA1 immunohistochemistry in part C of the diagram and EBRA in situ hybridization in part D of the figure. We can detect the virus in somewhere in the region of 5 to 15% of all cases of diffuse large B-cell lymphoma, where it predominantly is associated with the ABC type. Now, you'll see why in a few slides why the Epstein-Barr virus is important to, or at least has led us into some of the studies of the tumour microenvironment. But for now, it's important to recall that originally EBV positive diffuse large B-cell lymphoma was defined as EBV positive diffuse large B-cell lymphoma of the elderly. And of course, that's now changed in the most recent WHO classification, because we now recognize that EBV positive DLBCL occurs also in younger children. It was I think there's I guess a dogma in the field that the increased or the reported increase in the prevalence of EBV positive diffuse large B-cell lymphoma is associated with a decreased EBV specific immunity associated with senescence. However, and I'm not really going to talk about this today, some work that myself and a colleague in the University of Birmingham, Graham Taylor, have been doing recently indicates that systemic immunity to EBV is unaffected in people with EBV positive diffuse large B-cell lymphoma, reinforcing the possibility that the immune escape mechanisms are really situated in the tumour microenvironment itself. There's a reasonable amount of evidence that the virus contributes significantly to alterations in cell signaling in EBV positive diffuse large B-cell lymphoma. A frequently expressed virus protein is this one. It's the latent membrane protein 1. It is an oncogene. It's the EBV oncogene, which can mimic a constitutively activated CD40 receptor by virtue of the fact that it has these transmembrane domains that you can see on the left-hand side of the image, that enable spontaneous oligomerization of the receptor in the cell membrane, meaning that the receptor signals without the need for a ligand, in contrast, of course, to the normal CD40 signals, which require a CD40 ligand. So we were interested, and this is the starting point for the work that I'm going to describe to you. We were interested in looking for potential transcriptional targets of LMP1 that might help us to distinguish the impact of LMP1 and CD40. Remember, the if you like, the normal receptor counterpart of LMP1 in primary B cells as a way of trying to understand what LMP1 might be doing in EBV positive diffuse large B-cell lymphomas. So we performed a microarray analysis at that time, in which we either stimulated primary human germinal center B cells that we isolated from normal tonsils with CD40, or we introduced LMP1 into those cells, and we measured the transcriptional consequences. Not surprisingly, we found, as you can see here, we found a fairly healthy overlap in the transcriptional targets of CD40 and LMP1, which again would not be surprising given the similarity in the signaling mechanisms used by these two receptors. But reassuringly, we found some targets which were somewhat different. And when we compared those targets to genes differentially expressed in diffuse large B-cell lymphoma, we were able to identify a subset of 337 putative pathogenic targets of LMP1, which were not regulated by CD40. I'm not going to clearly be able to describe a good deal of those transcriptional targets and the pathways into which they fall, because that would take most of the afternoon. But suffice to say, at this point, a couple of really key pathways emerged, one of which we've spent a number of years working on. That's the sphingosine 1-phosphate pathway, which I'm not going to talk about today. And the other one, which I'm going to focus on, is collagen signaling. Just to really, I suppose, give you a bit of background, in my undergraduate days, collagen was really regarded as a structural component of tissues, but we now know, of course, that collagen is a very important signaling molecule that signals through a number of different types of collagen receptors. And the one that popped up in our analysis of being of particular importance is this one, highlighted in the red box. It's called discoidine domain receptor 1. Discoidine domain receptor 1 is a receptor tyrosine kinase that binds collagen and is activated by collagen, in contrast to a number of, or the majority of receptor tyrosine kinases, it already exists in a pre-dimerized and inactive form. But the binding of collagen to the receptor causes a conformational change, which enables autophosphorylation and the activation, the binding of other molecules, signaling molecules to DDR1, and the activation of a variety of different downstream signaling pathways. Now, just to show you that we were able to we were able to confirm that LMP1, but not CD40, upregulated the expression of DDR1 in primary human germinal center B cells. You can see the fairly sizable induction of DDR1 mRNA that followed the expression of LMP1 in primary B cells. We did not observe any change in the expression of DDR1 when we stimulated the same cells with CD40 ligand. Of course, the next thing we did was to look in diffuse large B-cell lymphoma, and we found a prominent upregulation of DDR1, not just in the EBV positive tumors, but in a significant proportion of tumors which were EBV negative, suggesting that there are non-viral mechanisms which can promote the overexpression of DDR1. And you can see here that on the top left, you can see a normal germinal center, which is completely devoid of DDR1 expression. And we can see expression here in 2 or 3 examples of DDR1 in the tumor cells of diffuse large B-cell lymphoma. Now, at this point, we decided to look at the nature of the collagen ligands that might be present together with DDR1 in these primary tumors. And we performed a meta-analysis of over 2,000 cases of diffuse large B-cell lymphoma using publicly available data sets. And what we found was a striking overlap between the collagen subunit gene expression and genes that were positively correlated but not negatively correlated with DDR1 in DLBCL. And these are the 14 or so collagen subunits that were positively correlated with DDR1 expression in DLBCL. And you can see that 4 of them come from collagen 6. And when we stained for collagen 6, unlike the other collagens that we also stained for, at least the majority of these we stained for, we only observe this fine reticular network of collagen 6, which appears to surround and is intimately associated with the tumor cell population. So collagen 6 appears to be a crucial collagen type that is overexpressed in DLBCL and is associated with DDR1 expression in those cases. Now, we've shown in previous studies that the activation of DDR1 is capable of rescuing lymphoma cells from chemotherapy-induced cell death. And when we looked at the expression data from our meta-analysis, we found a very striking association between protection from apoptosis and DDR1 expression, suggesting that we were seeing the same thing, the same phenotype in the primary tumors. One thing we didn't expect was a very strong association between the expression of DDR1 and genes associated or important for chromosome segregation, so that the higher the expression of DDR1, the lower the expression of these genes associated or important for chromosome segregation, shown here in a gene ontology analysis. And of course, chromosome segregation is crucial. Proper chromosome segregation is crucial to prevent the induction of chromosome imbalances, which can ultimately lead to aneuploidy. Aneuploidy is known to be associated with more aggressive disease in many cancer types, including diffuse large B-cell lymphoma. We performed a number of studies looking for an association between DDR1 expression and aneuploidy using different signatures of aneuploidy that have previously been reported in the literature. And in fact, we found a very striking overlap between all of the transcriptional signatures of aneuploidy that we were able to obtain from the literature and DDR1 expression. In fact, I think it's important to mention at this point that we also use signatures that had been used to separate out the potential inhibition of proliferation by aneuploidy, so that in other words, we still observe this relationship between DDR1 expression and aneuploidy when we use signatures that only defined aneuploidy and were not associated with reduced proliferation. Now, just a few, I suppose, a few slides just to say something about the mechanism responsible for this effect. So we used a number of our in vitro systems to explore if indeed we could see if we could see that DDR1 was associated with aneuploidy in our in vitro system. So the first thing we did was to look at a number of different regulators of chromosome segregation, and we found that DDR1 very robustly downregulated the expression of this molecule SEMP-E, which is a kinesin motor protein that is required for proper chromosome segregation. You can see in a couple of cell lines here, we see a nice downregulation of SEMP-E in the presence of collagen. These cells overexpressed DDR1. When we add collagen, we get an induction of DDR1 phosphorylation, which is associated with a loss of total DDR1 that's to do with the negative regulation of DDR1 expression itself once we activate the receptor. But the key observation here is that we see downregulation of SEMP-E, both at the protein level and at the RNA level, following the activation of DDR1 by collagen. We see that SEMP-E is also downregulated in diffuse large B-cell lymphoma. We see a nice nuclear expression of SEMP-E in normal germinal center B cells, and we see variable loss of SEMP-E in DLBCL tumors. SEMP-E inhibition, in fact, if one wants to induce aneuploidy in otherwise karyotypically stable cell systems, in fact, the best way to do this is to use a SEMP inhibitor. So the SEMP inhibitor is applied to a cell that induces the mitotic spindle checkpoint, which would normally arrest the cell in cycle. But if you then use the MPS1 inhibitor, which allows the cell to enter the cell cycle, then you can induce aneuploidy. It's a way that lots of groups use to induce aneuploidy in vitro. So SEMP-E has a pedigree as an inducer of aneuploidy. So I suppose the killer experiment is shown here. The question is, could we use DDR1 in vitro to induce aneuploidy in lymphoma cell lines? And the answer was yes, otherwise I wouldn't be showing you this slide. So we used a constitutively active DDR1 construct rather than collagen this time, but we see the same effect with collagen when it's added to cell lines overexpressing DDR1. But essentially, what happens in this experiment is when we add a constitutively active DDR1 receptor, we can induce aneuploidy, as you can see on the right-hand side here, compared to cells treated with vehicle, with the empty vector, or cells treated with a mutant form of the activated DDR1, which shows some DDR1 activity, which accounts for this slightly raised number of chromosomes compared to control. But the key take-home message here is in vitro, a constitutively active DDR1 is sufficient to induce aneuploidy in some, but not all, lymphoma cell lines. And that's something we're investigating at the present time, because it appears that the cells that in which DDR1 can induce aneuploidy may have certain genetic defects that allow the DDR1 expressing cell to escape the mitotic spindle checkpoint. But that is a story for another day. Okay, so I guess now really just to think about why this might be important. I think, you know, from our point of view, and we know pathologists have known for many years, and in fact it forms part of the WHO classification these days, that there is a very clear link between chronic inflammation and the development of lymphoma, and this could provide one mechanism to explain this link. One thing I haven't told you is that LMP1 the EBV oncogene is only oncogenic under certain conditions. And it may well be that the induction of DDR1 by LMP1 could be, if you like, a molecular switch that would allow LMP1 to switch from being a protein that is normally associated with the normal life cycle of EBV in people who are asymptomatically infected to become an oncogene that can promote lymphomagenesis. And I think some of the work that we've done in terms of identifying patients, different types of DLBCL patients with more or less aneuploidy could be important for stratifying patients for standard of care therapies. And the reason is, of course, that we know that aneuploid cells are resistant to certain antimitotic drugs. And I suppose on the back of this, we could also provide potential new opportunities to therapeutically target aneuploidy in cancers. One thing I haven't shown you that we've been working on is the transcriptional effects that aneuploidy induces in B cells. And what we see is something that's been described for other cell types in the literature, which is the induction of both energy and proteotoxic stress. Now, proteotoxic stress is induced by the increase in protein that follows the increase in genetic material in aneuploid cells. And that can lead to cell death. Now, clearly, cancer cells with aneuploidy have managed to survive any cell death that would have been induced by proteotoxic stress. But I think there's a lot of interest now in the potential to target that proteotoxic stress. In other words, to induce further proteotoxic stress, which can result in the induction of apoptosis. So, if you like, increasing further proteotoxic stress in an aneuploid cell could tip them over the edge and result in a therapeutic benefit. And finally, it might also be possible to target the collagen itself. There's a lot of interest in the present moment about targeting collagen synthesis, for example, in patients with severe COVID-19, but also in cancers that have a particularly prominent fibrotic component, for example, pancreatic cancer. A good example of this would be the antifibrotic drug pirfenidone, which is already approved for the treatment of patients with idiopathic pulmonary fibrosis. That's already in phase 2 cancer trials. So one can think of ways in which one might target the collagen as a potential therapy in patients with diffuse large B-cell lymphoma. So thank you very much for listening. I would just like to acknowledge a few important people in the work that I presented, particularly Matthew Pugh. Matthew's very kindly agreed to take some questions in my absence this afternoon, but also particular thanks to Sandra, now Sandra Davis, after having recently got married, Xiaohong Liu, Tracy Perry, Lauren Lupino and Zum Lakada, who were particularly responsible for much of the data that I've presented today. So thank you very much, and Matt will answer any questions you have.
Hi there. Hello.
Hi.
Yes.
I'm here. I'm actually here.
Yes, Paul. Thank you so much, Paul. It's great you could join us. That was most fascinating talk. I think we have a question from Mary Cahill. She says, I wonder, is there any data on tyrosine kinase inhibition of DDR1 in reducing collagen 4 interactions? Could collagen inhibition be related to the periorbital edema we notice when we use TKIs for treatment of CML?
On the second point, I've got no idea, but it's entirely possible. But the first point there is I think is highly relevant, because what we do know is that a number of the RTK inhibitors do in fact target DDR1, as DDR1's a recognised so-called off-target effect of some of those inhibitors. So I wouldn't be surprised at all if if those inhibitors, when used in patients, might also have might have a therapeutic effect through DDR1. Whether or not what we don't know is whether DDR1 itself is responsible for the increased deposition of collagens. That's something we're looking into at the present time.
Great, thank you. Eva has also commented. She says, "Great talk. Is there any reason why collagen 6 is most expressed or correlated with DDR expression?"
Collagen 6 is, generally speaking, collagen 6 is one of these. Unlike, let's say, if we take, say, collagen 1, which is one of the collagens that's been most studied in tumours. Collagen 1 tends to form the kind of dense fibrotic bands that you see in tissues in general. But if you think of tumours like nodular sclerosis, Hodgkin, we see type 1 collagen forming those big sclerotic bands that are characteristic of nodular sclerosis disease. And we do see type 1 collagens in DLBCL, but they tend not to be associated very closely with the tumor cells. Type 6 collagen is one of those collagens that forms that kind of reticular network, and what's interesting, I think, is that it comes up. So we've done a big expression study now of COVID-19 lung tissue. There was something like 120 samples from COVID patients. And of all the collagens, the three that pop up actually are exactly the same three that we think are most important in lymphomas, including collagen 6 and also collagen 3. So there is something unusual about these collagens. They pop up, as I say, frequently not just in diffuse large B-cell lymphoma, but also other lymphomas that we've been studying, as well as inflammatory diseases like COVID.
Great. We've got a question from Elisabeth Vandenberghe. Going back to GCB cell of origin, any difference in DDR1 expression in the different cellular origins?
No, that's a great question, Elisabeth. No, we don't see any difference. So both subtypes, there seem to be a subset of both ABC and GCB type that overexpress DDR1. I suppose what we have seen, which I think is of interest, and you touched on the importance of TP53 there. In what we do see is that we think that TP53 mutations are probably crucial in providing that anti-apoptotic signal that enables the cell to escape from the mitotic spindle checkpoint, and actually that observation is supported in part by our reanalysis of several data sets, where we only see an effect of DDR1 expression on somatic copy number alterations when P53 is mutant.
Fantastic. I think that's all the questions, Paul, but that was a really, really, really interesting talk. Oh, we've got one more. Hillary's just snuck in there. So we're not going to exclude Hillary. She says, have you looked at the role of lenalidomide in modifying DDR1 expression?
No, but that's a great question, Hillary. No, I mean, I suppose it's what we don't have are very good models, of course, of diffuse large B-cell lymphoma, either in vitro or in vivo. So it's kind of difficult to model what's happening in patients. And we have obviously, it's very unusual to have access to patients who've been treated and then been biopsied. So, but it's a great question. It wouldn't surprise me at all if it had an effect.
Okay, and I don't know if anybody's any other questions for Paul. Well, then that just leaves me to thank him so much, and thank you also for turning up live to answer all those very interesting questions. So thank you so much, Paul.
Philip.
And hope to see you in Limerick soon.
Thank you.
Good luck. Take care.
Definitely. Oh yes, we'll hope to. Yeah. Thanks.
Okay. Fantastic. Bye bye. So in fact, I think we're dead on time, and we've got our last speaker, who is Dr. Daniel Angelov, who's a hematology SPR, currently in pediatric hematology in Crumlin, got an interest in malignant lymphoid disorders, worked with Elisabeth Vandenberghe in St. James's Hospital, and he's going to talk a little about the St. James experience with transplant in cutaneous T-cell lymphomas. So I think we've got the recording, and then he's live after that.
Hi, my name is Dan Angelov. I'm a hematology SPR working in CHI Crumlin at the moment, and I'd like to thank the BCNI Symposium organisers for inviting me to speak at this meeting to present results of a retrospective analysis we carried out in St. James's Hospital on patients undergoing allogeneic stem cell transplantation for cutaneous T-cell lymphoma. The brief background is that cutaneous T-cell lymphoma is quite a rare disease. The approximate incidence is 0.4 cases per 100,000 per year, and the majority of these are mycosis fungoides patients. There are a smaller number of Sézary syndrome patients. However, in hematology, we probably see more of these than we do of the indolent mycosis fungoides. There's a slight male predominance, and the incidence is increasing over the past 3 decades of all forms of cutaneous T-cell lymphoma. The staging is different than our usual Ann Arbor staging we use in non-Hodgkin's and Hodgkin's lymphoma. So the staging is quite complex, and it's based around tumor stage, nodal stage, metastases, and blood stage, whether there's leukemic involvement or not. And clinically and practically, the way we divide these patients really is patch and plaque stage mycosis fungoides, or patch and plaque stage disease. And the majority of these patients will be managed under dermatology. Dermatologists specializing in the management of cutaneous T-cell lymphomas. And these patients may often, or are often managed using topical therapies as well as oral therapies. And you have the second group of patients who have tumor stage disease or erythrodermic disease. This can be erythrodermic mycosis fungoides or Sézary syndrome patients. And these patients will often be managed by clinical hematologists due to the more aggressive nature of their disease. Looking at overall survival and disease progression in both of these diseases, there's quite heterogeneous survival and disease progression data depending on the stage of the disease. So if you look at stage 1A, this will be plaque stage mycosis fungoides. These patients have an excellent overall survival at 10 years of approaching 100%. And as I mentioned, they have a very indolent disease that's managed on an outpatient basis by dermatology. Contrast that to patients with stage 4 disease, which would be all Sézary syndrome patients, and then erythrodermic mycosis fungoides patients. These patients have 100%, 70 to 100% risk of disease progression at 5 years and at 10 years. And so obviously different treatments are required for these different stage patients. So what we did was a single center retrospective study assessing consecutive CTCL patients undergoing first allogeneic stem cell transplant between the years 2010 and 2020. The conditioning regimens used were Cy-TBI, myeloablative conditioning for transformed mycosis fungoides in patients less than 45 years old. 2 reduced intensity conditioning regimens were used. The first one, Stanford protocol, was used in patients who were willing to travel to Northern Ireland. There they could receive total skin electron beam therapy, as well as coming back to St. James's, where they would receive total lymphoid irradiation and anti-thymocyte globulin. Mycophenolate mofetil made up part of the graft versus host disease prophylaxis in this conditioning regimen. For patients who were unable to travel to Northern Ireland, they received Flumel and antithymocyte globulin reduced intensity conditioning protocol. GVH prophylaxis was cyclosporin in all three regimens, as well as post-transplant methotrexate in the Cy-TBI regimen. We collected patient-specific data, including histological diagnosis, date of diagnosis, pre-transplant disease status, and prior therapies. We collected transplant data, as well as post-transplant outcomes. This included overall survival, progression-free survival, and incidence of acute and chronic graft-versus-host disease. We also captured chimerism T-cell receptor gene rearrangement data. Moving on to the results. I'm sorry this is a bit busy. This is a table of some of the results we collected. I'd like to bring your attention to disease stage. So you can see that 11 out of the 15 patients that we analyzed over the 10-year period had stage 3B or stage 4 disease. I'd like to also bring your attention to the fact that 11 of the 15 patients received systemic chemotherapy. So this was a group of patients who had advanced stage disease and quite aggressive disease biology. If you look at pathology, you can see that 8 patients had Sézary syndrome, while 7 patients had mycosis fungoides. 4 of these had transformed disease, so a more aggressive disease biology again. And there was a 2-to-1 males to female split, which is what is described in the literature for cutaneous T-cell lymphoma. Other therapies that were received by these patients included involved field radiotherapy, cyclosporin, infliximab, methotrexate, extracorporeal photopheresis, alemtuzumab and brentuximab vedotin, and bexarotene would be a skin-directed therapy, which would be delivered by dermatologists. It's an oral retinoid. So further results, I'd like to point out the 5 of the patients in the 15 cohort received subsequent donor lymphocyte infusions. And if you look at acute and chronic graft-versus-host disease, 13 of the 15 patients experienced grade 2 or higher acute graft-versus-host disease, and 5 of the 15 experienced chronic graft-versus-host disease, all of which involved the skin. CMV reactivation occurred in 5 patients, and I will show you the Kaplan-Meier curves next. So this is just an outline of what happened really in terms of survival data and relapse data in this patient cohort. So in summary, 15 patients underwent allogeneic stem cell transplant for CTCL over this 10-year period. 2 patients died within the first 100 days. These patients died of CMV pneumonitis, and another patient died of pneumonia. 13 patients were in complete remission at day 100, and 5 of these subsequently had relapsed CTCL. One of these patients who relapsed died of progressive disease, and the other 4 patients are all alive after either donor lymphocyte infusion or weaning of immunosuppression. 1 further patient died at 14 months due to pneumonia, and there are 11 patients alive currently in continuous complete remission. So if you look at the Kaplan-Meier curves for overall and progression-free survival, the overall survival at 5 years was 72.7%, and the progression-free survival at 5 years was 48.5%. You can see the drop-off initially with the early relapses and the treatment-related mortality. However, there's a plateau in both survival curves beyond the 2-year mark. Briefly, 2 of the patients who received reduced intensity Stanford conditioning had mixed chimerism, and 1 of the Flumel reduced intensity conditioning patients had a mixed chimerism at day 100. All myeloablative conditioning patients had full donor chimerism at day 100. The graft versus lymphoma effect is interesting in our cohort. Essentially, we had one patient who had DLI at 11 months for a small detectable Sézary syndrome clone in peripheral blood, and 6 years later, they're in molecular remission. One patient had a rapid wean of cyclosporin for disease relapse at 5 months and remains in molecular remission 2 years post-transplant. One transformed mycosis fungoides patient had a DLI with gemcitabine and remains in remission 6 years later. And 2 further patients had donor lymphocyte infusions for mycosis fungoides relapses post-transplant, and both remain in remission. To conclude, our data demonstrates that in carefully selected patients, allogeneic stem cell transplant provides durable remissions in cutaneous T-cell lymphoma. In patients that do relapse post-transplant, these patients can be salvaged with donor lymphocyte infusion due to an evident graft versus lymphoma effect in cutaneous T-cell lymphoma. We did note a high incidence of GVH, particularly of skin GVH, and this is likely due to the chronic breakdown of the natural skin barrier in these patients. And Stanford conditioning, which is a chemo-free regimen designed to reduce the incidence of acute graft versus host disease in our cohort, did not mitigate this risk. And the early treatment-related mortality in our cohort of 13% compares favorably with recent EBMT and CIBMTR data, where they demonstrated early TRMs of 19 to 26% in cohorts of 242 patients. I'd like to acknowledge the following people who helped with data collection and maintaining the transplant database. I'd like to thank you for your attention, and I'm happy to take any questions.
Hello, thank you, Daniel. That was really, thank you, Daniel. That was a lot of hard work you put in, and really well presented, and it's great to see that the results are so good compared to international standards. So well done. I'm not sure if anybody's got any questions for Daniel. But obviously really good results. I noticed brentuximab was kind of used very sparingly as initial therapy. That may be historical. Do you think that's been used more often in cutaneous T-cell lymphomas now?
Yeah, absolutely. Some of these patients, a lot of these patients were prior to the approval of brentuximab vedotin. So only 2 of our patients actually received it pre-transplant, and 1 received it post-transplant for relapse. So yeah, it was just purely down to the fact that it was before the time it was approved.
Okay, great, thank you. If nobody has any other questions, I think that brings this session to a close. And probably, I expect Eva would probably like to say a few words before we all go home for the weekend. Eva?
Sorry, my mute button was on. Can you hear me?
Okay, yes, we can hear you, Eva.
Yes. So first of all, I think a huge thanks goes to Dave, who works in the behind the scenes as our technician for the day. Dave, I'm sure that you learned loads about blood cancers. You didn't know there is so much we can talk about it. And I also like to thank everybody who stayed with us all day today. I think we had a great turnout. And I just want to remind everybody that all the talks have been recorded, and we will send out the links for these recorded in case anybody missed them and you would like to listen back to them. And I think just the last thing to say is that I really hope that next year around the same time, we will be able to meet in person. Thank you very much to everybody.
Thank you, Eva. Thanks, everybody. Good luck. Be safe.
Thank you. Bye-bye.
Bye.
Thanks.
That's all. Bye-bye.
Everyone.
Bye, everybody.